So, welcome everybody. I'm going to start 2 minutes of introduction. Thank you all for coming. This is our second annual conference in our series on social genomics. I'd like to acknowledge our generous funding from an R13 mechanism through the National Institute of Aging and COAL initiative funding from the Follette School of Public Affairs. It keeps the series free, and we can sponsor travel, especially for younger scholars. I'd like to recognize our co-organizers, 2 in the front here. So Lauren Smits, Corinne Engelman, James Lee, I saw somewhere. James? He was here. James will be back. And Q I see in the back. So the reason I'm highlighting, these are all chairs of the sessions and come from different disciplines. So I want to underline this fact that this is a very interdisciplinary conference, and we think that as you have a problem or a question with what you're doing, there's an expert here in the room. I think that's going to be relevant. So that's, I think, a really nice feature of the conference. I feel like this is, like thinking back, this is my favorite type of conference to go to as a junior scholar, where it's small enough where you can have one-on-one time with someone and introduce yourself, and please do that. This is a friendly audience of interdisciplinary scholars who are really willing to talk about collaborations or what problems you might be having with your work. So I really would encourage you to do that, where it's an intimate group that we're going to be having a lot of meals together and have beautiful weather to walk around here. So encourage that type of collaboration, and please introduce yourself. I'd like to thank Mindy and Tommy and Angela and the Follette outreach team for setting every single thing up and making this work seamlessly. And they were in charge of the weather, so thank you for that too. I'll say, so we've included, as you see in the program, sessions that cohere around topics that I think of general interest to everyone in the group, everything from GWAS methodology, G/E interactions, epigenetics, frontier issues that are outside those particular regular topics. So I think there's a lot of exciting work that we'll see in the next 2 days. So sit back and relax, and certainly meet many people in Midlife. I think that's the real core, is that we have a lot of local expertise, co-chairs across disciplines, and I hope you'll come up and introduce yourself and let us get to know you. So with that, I'll ask the 3 session 1 speakers to start making their way to sit up front here. And Corinne Engelman's going to introduce the first session. While Corinne's getting set up, Wi-Fi, the thing I missed. So there are, on many tables, instructions in this little placard, in this little card. So let me or Mindy know if you're having problems, but otherwise we should be set. There's 2 sets of restrooms on this floor, and we'll have a lot of our meals in this area. And with that, I'll hand it over to Corinne.
Hi, I'm Corinne Engelman. I'm a professor in the Department of Population Health Sciences. I'm a genetic epidemiologist by training, and my research is with Alzheimer's disease and GWAS and polygenic risk scores. Let's see. I'll just get off and I'll start off introducing the session. We're gonna, if we have time, we'll take 1 to 2 questions at the end of each presentation. If we run out of time, we'll probably hold them till the end, but that's the goal. And when we do take questions, we'll make sure that we use the microphone, because this area up here is mic'd, but the audience area is not, and there's a recording of this. So we want to make sure that everything is heard. So genome-wide association studies, or GWAS, have been performed for the past 2 decades, and results from GWAS, and especially meta-analyses of the GWAS, have increased our understanding of disease and traits and allowed us to more accurately predict the risk for disease or the value of a quantitative trait. But there's still room for improvement in GWAS methods, and that's the theme of this session. So our first speaker will present a new method, genetic-related matched association study that builds on the strengths of population-based GWAS and family-based GWAS. Our second speaker will present a principled and machine learning-assisted statistical framework for estimating individual genetic effects that model complex and nonlinear interactions between high-dimensional genetic data and environmental factors. And then our third speaker will present results from a study of mother-child dyads to determine whether maternal adverse childhood experiences are associated with increased inflammatory gene expression and whether a parenting intervention moderates this association. So we can start off with our first speaker.
Oh, is someone else starting first?
Different slides.
Cool. Okay. Hi everyone. I'm Dhruva. Today I'll be talking about how genetic relatedness matched association studies can increase diversity, reduce bias, and increase power relative to traditional GWAS. So I'll be talking about what a GWAS is, what genetic relatedness matched association studies are, how a GRAMA study works, some preliminary empirical results that we have, and future work that we have planned using this. So a genome-wide association study is a study where we want to find an association between genotype alleles and phenotypes of interest. And so what we typically do is run a regression of a phenotype on some SNPs and some controls such as PCs. However, there are some problems with traditional GWAS, and one of the main ones is bias from population stratification. And so population stratification is a situation where you have systematically different allele frequencies in different subpopulations, as well as systematically different phenotypes in these subpopulations. And so the way that researchers typically deal with the stratification bias is to subset their sample to continental ancestry samples. However, this restricts genetic diversity and ends up discarding a lot of the data that you could be using in a GWAS. Even after subsetting to these continental ancestry samples, which is typically the European subsample, there are other residual forms of bias that can creep into the estimate. Another method that people have started using to control for this bias is doing a sibling GWAS, where you subset the sample to sibling groups, and conditional on siblingship, genotypes are randomly assigned. And so if you control for these allele frequencies, you can get estimates that are unbiased. However, because you're subsetting the sample just to the sibling groups, you end up getting much smaller sample sizes with lower powered estimates. It's not going. Thanks. So GRAMA is the method we propose to overcome these shortcomings of traditional GWAS. And so similar to how in a population GWAS, you would find the effect of an additional allele on a phenotype relative to the population mean, and in a sibling GWAS, you would find the effect of an additional allele relative to the sibling mean. GRAMA makes these comparisons relative to a relatedness group mean, where the relatedness group is formed by matching people who are as genetically close to each other as a threshold value of kinship set by a researcher. And so because you can set these kinship values at levels corresponding to, for example, full siblings or first to nth degree relations, up to population GWAS levels, GRAMMA becomes a generalized model of sibling and population GWAS. So here's a quick visualization to show you how GRAMMA works. So consider the entire population. Individuals that are closer to each other are also more closely genetically related and have a more recent common ancestor and more similar allele frequencies. In a traditional GWAS, you would only use this smaller, continental ancestry subset, and so you're discarding a lot of the data that you could be using. But GRAMMA avoids having to subset to these ancestral subsamples, and instead can use the entire sample. And so suppose we set the threshold value at the level of siblings. In this case, we could go through each individual in the population, match them to their sibling group, find within group effects, and average these effects out over the population. So here, individuals without siblings are dropped, and so it's equivalent to a sibling GWAS. And so we get unbiased estimates, but as you can see, we've discarded a lot of the data in our sample, and so we have small sample sizes, and so lower powered estimates. If we relaxed this relatedness threshold and made it more permissive up to the level of first cousins at third degree relation, you can see that we have larger relatedness groups and more relatedness groups. And so although some bias is creeping into the estimates, we also have these larger sample sizes, and so more power to estimate direct effects. And so from this, you can see two main advantages of GRAMMA. There's no need to limit the sample, and so you can use these more diverse samples and underrepresented ancestries that are typically discarded in a GWAS. So more recently, people have started to try to use these diverse samples by running a GWAS in different ancestral subgroups and then meta-analyzing these estimates. However, PCs have been shown to be, or it's been shown to be less effective to use PCs in smaller subsamples. And even if you did this meta-analysis, 10% of the samples in the UKB aren't cleanly assigned to an ancestral subgroup, and so you still aren't using as much data as you could be. And so the implication of using more data is that you have larger sample sizes, and so you have increased power to estimate direct effects, and all of this will lead to improvements in genetics research down the line. And so GRAMMA can also shed light on this bias variance trade-off across the relatedness thresholds, and so what you can see is that, at kind of high relatedness thresholds, so the one corresponding to full siblings, as I said before, there's no bias in these estimates, but you have smaller sample sizes and so lower power. And as you make this threshold more permissive and go out to higher degree relations, you get more bias, but you have larger sample sizes and so more power. And GRAMMA can actually get a better bias variance trade-off than population GWAS because holding some amount of bias fixed, you can reduce the sampling variance by using more of the sample. And so you can imagine that at a low enough threshold, one of the relatedness groups is roughly equivalent to the European subsample. But since you're still using the other subsamples, bias is at most what it would be in a GWAS of these separate subsamples, but you're using more data, and so you have more power. We also have some preliminary empirical results, having used GRAMMA on some UKB data. And so we did this with 400,000 individuals of white British ancestry. We ran King relationship inference software on this sample and excluded samples of poor quality genotypes. And we did this with 5 degrees corresponding to full siblings and first to fourth degree relations. And we also compared this to a GWAS using only PCs as covariates. We did this for 3 phenotypes: BMI, height, and educational attainment. So first we looked at power in terms of the mean chi-squared statistic. And so here we have a graph of the mean chi-squared statistic across all the degrees for the 3 phenotypes. And below this we have a table with the effective sample sizes after adjusting for the fact that we're dividing the sample into relatedness groups. So what you can see is there's, and so we can use this mean chi-squared statistic -1 as a proxy for power. And what you can see is there's a clear upward trend across the degrees as you make it more permissive and increase the sample size, showing that you have more power with higher sample sizes. We also looked at bias from correlation and heritability. And so here we have the genetic correlation with the full siblings trait across the different degrees and phenotypes. What you can see is, so if correlation was much less than 1, then it would be an indication that there's some bias in your estimates. Here, all the correlations were statistically indifferent from 1. However, you can see that the standard errors are quite large, and so this imprecision could be masking actual differences in the correlation across degrees. Finally, we looked at heritability, and so which could also show more bias. And so here, BMI seems to have roughly the same heritability across the degrees, but we do see higher heritability relative to the full siblings trait for height and educational attainment, which could indicate that some bias is creeping into the estimates at these more permissive relatedness thresholds. And so, yeah, in terms of future work, we're going to be doing some further empirical exploration, and we'll be making some computational improvements, including additional input filters and adding covariates and faster processing times. Yeah, I'd like to thank Patrick for being an excellent mentor and architect behind GRAMMA, John for his coding expertise and detailed feedback regarding GRAMMA code, and the SSGAC, especially Dan and Alex, for helpful discussions. And yeah, thank you for listening. If you have any questions, I'm happy to take them.
We definitely have time for a few questions.
Thank you. Thanks, Drew. This is super interesting. So one thing I'm wondering about is that when we use within sibling or family fixed effects, we're controlling for a lot of things, right? But like the shared environment within the family, and when you relax that threshold, you're no longer controlling for shared family environment. Is that correct?
Yeah. So.
Sorry, I'll just complete my, because
Oh,
I
yeah.
just wanted to follow up. So now, the fact that you're not seeing much bias there, it makes me wonder that what are we doing when we're doing family fixed effects? Are there, like, maybe the things that we think of as family environment, maybe those are not that important? It's actually just controlling for ancestry at a very, at a much closer level, approximate level than PCs could. So, yeah, if you could help us think through that.
So, in terms of different environments causing bias, so these would show up in the heritability estimates. And so, if you had different environments across these different degrees, that's where the bias would come from. And so in this case, this kind of does indicate that more bias is creeping in at these more permissive relatedness thresholds. And yeah, so actually we are kind of seeing those in this case.
It is much smaller than one would think, though, like for
Yeah,
even before.
In terms of these results, they are quite preliminary, and so these may not be the same as like what our final results are going to look like, but yeah.
Time for one more question?
Thanks very much. Just a technical question. So I find figures like this very informative. You can do stratified analysis and see if certain metrics change when you release, relax the threshold. I wonder if you just include everybody, if there are a parameter in the framework that can help you quantify, for example, say whether the heritability changes, between very stringent within sibling analysis versus population-based analysis. So if there comes a continuum of this parameter you can test in the framework for this type of parameters instead of doing stratified analysis.
If you can, so which parameter so you wanted to test the heritability?
For example, if we're wondering whether sibling-based analysis and population-based analysis will have different heritability. Mm-hmm. In your framework, is it possible to simply include everybody, but then you have the genetic relatedness between every pair of individuals in the framework? Is there a way to quantify this type of change of heritability within this framework that does not exclude any individual, but then you can test whether there is a trend of this change?
So I guess that would be dependent on every individual having a relation of the required type, and so like for the full siblings, if you wanted to include everyone would have to have like a sibling. But at each degree, in the first step, everyone is included, and then it's subset to just the people who have siblings. So this would be testing like the entire sample, except you're just subsetting to the siblings before finding the heritability parameter.
Yeah, thank you. I'm happy to follow up on that.
Okay,
I guess just very general thought is we're interested in doing some kind of a gene-environment interaction, but here the environment is not quantified at the individual level, but it's rather defined for pairwise relationship of individuals. But then I was simply wondering if you have a solution of how to quantify how genetic effect changes on that kind of direction in a framework. But I don't have an answer to it myself. Thank you very much.
Okay, yeah, we should definitely talk about that afterwards. Yeah, I might have misunderstood your question, but yeah, we can talk about that afterwards. Okay, great.
Okay, before we get to our second speaker, I apologize. I looked at the wrong abstract for the third presentation. I was wondering why it didn't quite fit the theme, but I was trying to make the best of it. So it's our third talk will be assessing biases in GWAS using family history across diseases and cohorts, a systematic evaluation. So much more in line with this session. Go ahead, yeah.
Okay, so hello everyone, I'm Jia Chen. I'm a fourth year PhD student in the biostat department. So today I will be talking about our most recent work on how to do valid GWAS on machine learning imputed outcome. So GWAS, genome-wide association study, is to identify the genetic associations with complex traits and diseases. And statistically, it's just a linear regression that regress the trait value or disease status on the genetic variations. However, even with the extensive biobanks, we typically lack the high-quality phenotype data that we can measure whether there is any genetic association with this valuable trait. For example, in the UK Biobank, the X-ray and MRI imaging data have a missing rate over 90%. So this limits our ability to identify the genetic associations with these valuable traits. And to address these questions, researchers have come up with a new study design called machine learning assisted GWAS. The idea is they use some variables that are easy to measure to impute these phenotypes that are difficult to measure, and then they directly do a GWAS on this imputed outcome. So this study design has been extremely popular in recent years. However, all these papers treated the imputed outcome, imputed phenotypes as observed, and totally ignoring the uncertainty in these machine learning predictions. So in our paper, we demonstrated this is a pretty bad idea in fact. So we did two GWASs. The first is we do a GWAS on the ground truth type 2 diabetes diagnosis. And then we also do a GWAS on the machine learning imputed type 2 diabetes. And we found that over 80% of the associations with imputed type 2 diabetes cannot be replicated using the ground truth type 2 diabetes. And we come up with a theory to explain this very high replication failure rate. So we checked our machine learning imputation model, and we found that the HbA1c, hemoglobin A1c, is the strongest predictors of the imputed type 2 diabetes. So the basic idea for hemoglobin A1c, so it is a measurement of how much of the glucose is attached to the red blood cells. So you can imagine for if the person have a type 2 diabetes, the body will fail to maintain control of the normal range of the glucose, then that person will have an elevated HbA1c level. However, as you can see, it is not only about the glucose, it is also about the red blood cells. So you can imagine if a person genetically have more red blood cells, then that person will also have an elevated HbA1c level. And so our hypothesis is that the genetic associations that is related to the non-glycemic, that is the properties of these red blood cells, and these genetic variants are not in fact associated with the control of the glucose, are associated with the imputed type 2 diabetes. And this will become we call it false positive associations or non-replicable signals on the imputed type 2 diabetes. And indeed, we checked the top associations in our imputed type 2 diabetes GWAS, and we found that all of them are associated with HbA1c levels. And if you look at the bottom of this panel, these are the traits related to the properties of the red blood cells. So as you can see, all of them are also associated with the properties of the red blood cells. However, none of them is associated with the ground truth type 2 diabetes, which is the second row here, and also related to how body controls the glucose, such as the fasting glucose level, which is shown on the third panel, third rows in this panel. So this confirms our hypothesis and shows that if you just ignore the uncertainties in these machine learning predictions, you are likely to get a lot of false positive associations using the current study design of the machine learning assisted GWAS. So to address this question, we propose a simple framework called post-prediction GWAS. So it's called POP GWAS for valid and efficient machine learning assisted GWAS. So compared with the conventional approaches that directly do a GWAS on the machine learning imputed outcomes, our framework will do two more GWASs. And the key idea of our framework is we will use the difference between the GWAS of the imputed and observed outcome in the label data. That is the data which we have the high-quality phenotype measures to debias the conventional machine learning assisted GWAS. So the intuition is that suppose in the label data, the GWAS of observed and imputed phenotype is exactly the same. So this is saying that the imputation is great. We can just completely ignore the uncertainties in these imputations, and we can trust the GWAS of the imputed outcome. However, if the results from the label data tells us that GWAS of imputed and observed outcomes differs a lot, then we need to debias because the GWAS of imputed outcome may not be the GWAS we actually want to do. So this is our POP GWAS framework. And to provide further intuitions, POP GWAS is data adaptive. So we can consider two simple scenarios. The first is we have the perfect imputation. That is, we can have magic machine learning algorithms to help us impute these missing outcomes. And then POP GWAS will degenerate to the GWAS of imputed outcome using the full data, that is, both the labeled data and the unlabeled data to do the GWAS. Because here, if we have perfect imputation, we can directly trust the imputations, and we will use the full information. However, let's say you have a terrible imputation. You just have a random noise in the imputations. Then pop GWAS will degenerate to the GWAS of observed outcome only in the label data. So this is saying that we will totally ignore any information that is related to the imputed outcome. So this is the intuition of pop GWAS. And moreover, pop GWAS have several key features. It is robust to any machine learning model you want. You can use linear regression, deep learning model, or even large language model. You can use any machine learning you want to impute these outcomes. And moreover, we show that it is the statistically optimal solutions for valid machine learning assisted GWAS. So this is saying that you cannot propose a more efficient machine learning assisted GWAS without sacrificing the validity of the GWAS. Moreover, it is very easy to use. It only requires summary statistics as input. As you can see, here is our script, and you only need to input 3 GWASs into our script, and our software will return you a machine learning assisted GWAS. And last, it is very fast. It can complete analysis for like 10 million variants within 3 minutes. So we showed that pop GWAS works pretty well in simulations. So here, different color represent different method, and the red color represents the GWAS only using the imputed outcome, and the blue color represents pop GWAS, and the yellow color represents the GWAS only using the label data, that is without any machine learning models. So as you can see in the panel A here, only using the imputed data will give you biased results. And in the panel B, it will give you a lot of false positive associations. And both pop GWAS and the GWAS only using the high quality measured outcome is unbiased and maintains the type error pretty well. But if you are at the panel C, panel D, and panel F here, as you can see, pop GWAS is more powerful than the GWAS without using any machine learning imputed outcomes. Moreover, pop GWAS is more powerful with more accurate machine learning models and more unlabeled data. And we also tried pop GWAS in our type 2 diabetes benchmarking examples. So we showed that all the pop GWAS associations can be replicated in the ground truth type 2 diabetes. And moreover, all these non-glycemic associations are not significant in pop GWAS. So demonstrate the ability of pop GWAS to maintain the well-controlled type error rate with the use of machine learning imputed outcome. And moreover, since we have demonstrated pop GWAS improves the GWAS only using the label data, we also propose power and sample size calculations that can incorporate the machine learning predictions. The idea is that in the classical power and sample size analysis, people only count how many data they actually measured, and they will just ignore all this data set with missing outcomes. However, here, what we are proposing is that you can also incorporate this data set without measuring the phenotype, but you have a machine learning model to impute these missing outcomes. And for example, if you want to calculate the power for a GWAS, and you only need to input the sample size of the unlabeled data, and also the sample size of the labeled data, and the accuracy of the machine learning algorithms, then it will give you a power. And we apply our method to bone mineral density, which is the primary indicator of the fracture risk in the clinic. And as you can see here, pop GWAS identifies around 40% more genetic associations with the bone density that is measured using X-ray imaging and with the help of machine learning imputations. And a lot of them can be replicated in a GWAS with an independent cohort. And we also identify a head-specific association that is only significant in head, but not on any other body sites. And it is verified in mice experiments. So far, I have talked about how to do GWAS on valid GWAS on machine learning imputed outcome, which is a linear regression. However, there are many more statistical tasks that people are maybe interested. For example, logistic regression, quantile regression, just a lot of other tasks. And for this task, the classical analysis, we're only using the label data to do the analysis. And what we are doing here is we are generalizing our pop GWAS framework to a new framework called PSPS. So it can not only deal with linear regression, but almost all the statistical tasks you can imagine. So the analysis pipeline is the same. You have 3 different data sets, and you need to apply the same data analysis pipeline to these 3 different data sets. And you will get 3 point estimates and their variance covariance matrix. And input these summary stats with our software, and it will give you a machine learning associated analysis. And I should note that our framework is not only data adaptive, but it's also user adaptive here. So if you are a cat person, our method's name will become PSPS, will become PSPS. So, yeah, so feel free to try this, and it will adapt to whether you are a cat person or not. So due to time limit, I will go through it pretty quickly. So basically, we show that PSPS can be easily applied to a lot of statistical tasks, including but not limited to interaction, quantile regression, instrumental variable regression, and generalized method of moments. So I will just use panel A and F to demonstrate the message here. So the message is that if you are using only the imputed data, you will get false positive results. And if you are comparing the PSPS with the classical method without using any machine learning, you will get higher power. So that is the take-home messages. So our software is publicly available on GitHub. We have the pop GWAS, which is specifically designed for GWAS. So only 3 summary stats as input. And we also have a PSPS. So basically, 3 point estimates as input and the variance covariance matrix, and then it will return a machine learning associated inference results. So to summarize, we demonstrate the risk of pervasive false positive associations for the current machine learning assisted GWAS, and we propose a simple solution, pop GWAS. We also generalize the pop GWAS to PSPS to deal with much more range of statistical tasks, and then the preprint of this method is available. So finally, I would like to thank all my co-authors, and especially Hugh, who mentored this project. I would also want to thank for the members of the Lab and the initiative in social genomics for very helpful discussions. And that's all I have, and thanks for your attention.
Time for one short question.
This is really interesting work. Good job. I had a question of how, if you thought about how your approach would compare to doing something like MTAG or Genomics SEM to combine a GWAS of the imputed phenotype versus the, I think you call it the ground truth phenotype. Mm-hmm. Just looking at the equations, it looked like you maybe get similar things by combining them using a method like that. Have you looked at something like that?
We have the mathematical derivation to show that these factor models will result a linear combination of 2 GWASs, but it is non-negative linear combinations. So it is like non-negative linear combination of the GWAS of the imputed outcome and observed outcomes. So if one part of the GWASs is biased, then this combined estimator will also to be biased of the ground truth GWAS.
Thank you.
Okay, we'll have our third speaker come up. And I wonder if maybe, since we're a small group, could we have whenever we talk, either as a presenter or asking a question, would it be okay if we said our name and where we're from? I think that would help us get to know each other. Okay.
Morning, everyone. My name is Yu Chang. I'm an assistant scientist at UW Medicine, working with Q. Today I'd like to talk about pervasive biases in a very popular study design called GWAS by proxy or GWAS. So in this talk, I will demonstrate how these biases will lead to erroneous results for Alzheimer's disease and other diseases and across different cohorts. So first of all, I'd like to acknowledge everyone who contributed to this project, especially the students in our lab who made a substantial contribution to this project, and also Jason and Qingshi who supervised this project, and also our local social genomics research group for very helpful discussion and feedback. Okay, so GWAS by proxy or GWAS is a new study design proposed in 2017. In a very traditional case control GWAS, we compare clinical diagnosis cases versus controls. Where in a GWAS, we compare the proxy cases versus proxy controls. So basically, instead of looking at the case control status for the participants, we look at the case control status of those participants' relatives, for example, their parents. So therefore, the proxy cases is defined as those whose parents or relatives are infected individuals, and the proxy controls are those whose relatives are not affected by disease. So using some mathematical derivation to show that GWAS can be used to estimate the same parameter as that in a GWAS, which is the SNP effects on disease status. So the motivation for GWAS is to boost the statistical power because it's usually very difficult to recruit enough cases, especially for the late onset disease such as Alzheimer's disease. So in this paper, they demonstrate that GWAS can indeed boost the power by replicate a lot of top findings in GWAS. For example, in this Alzheimer's disease, in the first scatter plot, where each dot is a top SNP identified in GWAS, the x-axis shows the effect size estimated from GWAS. The y-axis shows the effect size estimated from GWAS after some transformation. You can see that their effect size are very highly correlated, indicating that GWAS can indeed replicate the top SNPs. And also similarly, you can find that for coronary artery disease and type 2 diabetes, they could all replicate the top findings. So this what convinced people that GWAS is a really good idea to boost statistical power. So since then, it's become increasingly popular, especially for the AD field. But also, since then, this genetic field also moved from find the top SNPs to genome-wide analysis. So here in this paper, it shows that GWAS is a good idea for the top SNPs. So is this still a good idea if we do genome-wide analysis? So it's not enough study on that. So therefore, we conducted the GWAS for Alzheimer's disease using UK Biobank. And indeed, here, if we compare the top SNPs, indeed, it could replicate the top SNP. For example, the Manhattan plot between GWAS and GWAS looks very symmetrical to each other. And if we compare the effect size for the top SNPs, they're highly correlated. However, when it comes to the genome-wide analysis, we find a lot of biases. For example, in the panel A, we show the genetic correlation between AD and education attainment. So for the AD GWAS results, which is indicated in black, has a negative genetic correlation with education. However, the AD GWAS, which is shown in red, has a positive genetic correlation with the education, so which is very counterintuitive. The green one, which is a meta-analysis of GWAS and the GWAS, just lies in between. So panel B is a PRS application where we computed PRS for AD, and then we use that to predict cognition in HRS. Again, using AD GWAS to compute the PRS, it shows negative association with cognition. However, for AD GWAS PRS, we find no result. And again, the meta-analyzed result lies in between. So the panel C shows the Mendelian admission analysis where we assess the causal effect of education on AD. Using AD GWAS as input, we find that education is a protection factor against AD. However, if we use AD GWAS, again shown in red, we find that education actually causes AD, which is very counterintuitive, although it's not statistically significant. Again, the meta-analyzed result lies in between. So from this plot, we can see that when it comes to genome-wide analysis, there's a lot of divergent results between GWAS and GWAS. So if we believe that the GWAS is the ground truth, that means the GWAS has a lot of biases. What's more concerning is that it seems that AD genetic field is not aware of this bias. In these plots, we collected the large AD studies published in the last 10 years. We computed their genetic correlation with education and colored by different study designs. Again, the black shows the AD GWAS has negative genetic correlation with education. The red, which is AD GWAS study design, has positive genetic correlation with AD. And the green one is meta-analyzed the results, is lies between. And all the recent large AD studies, they are all meta-analyzed the GWAS and GWAS. And they stopped releasing summary stats for the GWAS and GWAS separately. They only released the meta-analyzed the summary stats, indicating that they are not really aware of this problem. Okay, so having found that there's a lot of divergent results between AD GWAS and GWAS, the natural question is, what are these biases? What causes this bias? Is there a way to isolate this bias? So it turns out we could do this using a study design called GWAS by subtraction. Basic idea is we have GWAS summary stats, we have GWAS summary stats. We just regress out the GWAS components from GWAS, and the remaining one we consider as the bias component or the non-AD factor. And we consider that as the GWAS for the bias. And for this setting, it turned out that we could do this analytically. And we developed an R package called GSUB that can be used to do this. So after obtaining the summary stats for the bias components, we'd like to know the genetic architectures for this bias to give us insight of what are these biases. So we computed genetic correlation with a lot of traits. And here, in this plot, I only listed the traits that have a significant genetic correlation with the non-AD factor or the bias component, which is shown in red. As a comparison, I also included the results with the AD factor shown in black. As you can see that it's positively correlated with the family history participation, indicating there's participation bias. It's negatively correlated with coronary artery disease, indicating there's a survival bias. And also negatively correlated with the not knowing parental illness, indicating there's a reporting bias. So having identified these biases, our next step is to remove these biases and to see whether we could get improved results. So for each biases, we explored two different ways to remove the bias. For survival bias, we followed a Maroney et al. Approach, where we limit our GWAS sample to those whose parents are at least 65 years old, which is onset age for AD, and also we further add their parental age in the regression covariates. An alternative approach is Yang et al., where we created a continuous disease risk based on the parental AD status, parental age, and AD prevalence in the population. Then we ran a GWAS on this created outcome. For participation bias, the first approach is just do a weighted GWAS, where the weight is constructed following this Follette et al. Paper. Our alternative approach is GWAS by subtraction, because we have a GWAS on participation. So we just remove the participation components from the GWAS and then use the remaining one to see whether we could get the improve the results. For reporting bias, our first approach is to include those who select I don't know in the survey question as controls. The rationale is that if their parents have AD, probably they should know. If their parents do not have, if they answer I do not know, most likely means that their parents do not have AD. So we include those as in controls. Another approach is, again, it's GWAS by subtraction application. We have GWAS on I don't know, then we just remove that component. So after that, here is the result. It turned out the Yang et al. Approach works best, although it's designed to remove the survival bias. It seems like it could remove other biases as well, depending on if we only look at the results with education, because after using the Yang et al. Approach, it gives a very similar genetic correlation with education as that given by the AD GWAS. So we find there's a lot of biases in AD. So the natural question is, how about other family histories and in other cohorts? Could we see the similar patterns? So we conducted GWAS for other family history in the UK Biobank and also in all of us, because in all of us it also asks family histories. So here's the result, which shows the genetic correlation with, again, with education. So we conducted GWAS for 10 diseases, then we compare with their respective GWAS result. So except the last two, lung cancer and COPD, all the other 8 diseases show some kind of divergent results. So for example, for prostate cancer, the black one shows the GWAS has no result with education, where the GWAS has positive genetic correlation. For high blood pressure, you can see that the GWAS result, which is shown in green, has a positive genetic correlation, indicating GWAS may have its own patterns of bias. And again, for example, for severe depression, the GWAS and GWAS looks different. For heart failure, heart disease, the GWAS looks different. And again, for diabetes, the GWAS looks different. So all this indicate that the biases are very pervasive across other diseases and cohorts. And to understand what are these biases, we again use the GSUB application and then calculate the genetic correlation with other traits. We find that for, first I could not see, prostate cancer, there's a participation bias and survival bias. For breast cancer, there's a participation bias. And severe depression there's a survival bias, participation bias, and the reporting bias. And the stroke, again, participation bias and survival bias. So to conclude, we identified biases in AD-GWAS leading to erroneous results for a genome-wide analysis, such as genetic correlation, PRS prediction, and Mendelian randomization. And this bias in the GWAS seems to be pervasive across disease and cohorts. So therefore, extreme caution should be exercised when utilizing GWAS results. And also, our study has some limitations. First is we treat the case control GWAS as gold standard, which may not be the gold standard. And also, it's unclear what metrics should be used to benchmark the performance of GWAS. Here we use the genetic correlation with education, which may not be the only metric. Maybe we should also consider other metrics. So that's all. Thank you very much.
Thanks for an interesting paper. I am a little wary of putting I don't know into the controls, and I wonder if you could expand a little bit, perhaps with some data in terms of that, given that when you're looking at other sources of bias, it gets rather specific. Blood pressure, heart failure versus heart disease. So it seems to me that, well, let me just pose the question about the control.
Yes, so again, so our rationale to put the I don't know group as control is because we, for example, for AD, we feel like if their parents have AD, probably they should know. If they say I don't know, maybe that means their parents do not have AD.
But do you have any data from others or yourself in terms of the size of the effect of doing that?
Yes, I did not show in this plot because by doing that, there's not much improvement in the results as in terms of genetic correlation with education. There's no change.
We have time for one more question.
This is interesting. Thank you. So my question have to do with the dealing with the survival bias. You know, I think people who are more educated live longer, so there's so there's a competing risk. Right, they have more time to to get these diseases. Mm-hmm. Right, so that's in part why we think that there might be this positive correlation with education. Right. So I was wondering, in order to, have you thought about using some sort of competing risk model to try to address that? Do you have, I guess, the question is, do you have the date, the age at which parents died in this?
Yes, so when we use that Meroni et al. Approach to correct for the survival bias. We limit their parents at least 65 years old. That's when they are still alive, is at least 65 years old. When they die, is die past that age.
Right.
When we add their age in the covariance, is either current age or age at death. So to some degree, we controlled for that.
Right. There might, yeah. I don't know exactly how, but there might be more, using the exact date as like in a more formal competing risk model might be possible in order.
Yeah, that's possible.
Yeah,
We
I
did
don't know.
not explore.
Yeah, we can talk about it.
Yes.
I think it's interesting. This is something we see in our own work also, so
Mhm.
I'm trying to figure it out together. So.
Yeah, sure. Yeah.
We actually do have time for one more question. I think Wenman had her hand up at the same time.
Thank you. Very nice study. I just want to ask, is it comparable with the pop GWAS we just learned?
Is that what?
Thank you for the. I just shout. Is it comparable with the pop GWAS we just heard about?
Comparable?
To the pop GWAS, the last talk.
Yes. So
Because it also sounds like an imputation-based.
yes, you could think of this parental history as kind of machine learning algorithm to predict your the outcome. So we could use pop GWAS to apply to this. The only problem is, for pop GWAS, you need to have some kind of ground truth as your good standard. For here, that means you need to have some samples. You know their ground truth of AD case control status. You also know their parents' AD status. But here we only use UK Biobank data. We do not know. There are some samples, but the sample size is very limited. So we actually used the pop GWAS. The result didn't look very well. So we suspect it's because of the small sample size.
Just a quick follow-up. Is it because you cannot accurately estimate the R in the pop GWAS?
Yes, the imputation quality is very, very low. Yes.
Thank you. Okay, if we could have the speakers for the next round, the next session come up, that would be great. Thank you.
All right, well, we're going to go ahead and jump right into the next session. It's great to see so many familiar faces in the room. For those of you who I do not know, my name is Lauren Schmitz. I'm a soon-to-be associate professor at the Follette School of Public Affairs here at UW-Madison. In my own work, I use genetic and epigenetic data to investigate how social factors affect health and health disparities across the life course. I'm an economist by training, but then went on to get additional graduate training in human genetics, and so I really enjoy bringing methods together in these fields. Yeah, we have a really exciting panel today. This is our first epigenetics panel, so we're going to be shifting over now into thinking about DNA methylation and uses of DNA methylation data. I'm going to go ahead and just jump right in. Our first speaker is Dr. Erin Dunn from Purdue University, and she's going to be presenting her research on identifying sensitive periods for the effects of childhood adversity on DNA methylation. Welcome. Thank you. Yeah. You guys know you have 12 minutes, and so I'll tell you when you've got 2 minutes.
Oh, wait, that's the mic. And then push to advance. All right, perfect. Awesome. So nice to be here. Thank you so much for organizing this. My talk is going to be different from the prior talks in that I'm going to give more of a bird's eye view about all of the work that's going on in my lab. I am literally 3 days into a new role at Purdue University. So I was at Mass General in Harvard for 12 years and just left, started officially on June 1 for a new position as professor of sociology, starting to lead a new initiative focused on sociogenomics. And the goal of what we're trying to do is to build a new university-wide center focused in this space. Our goal is ambitious, as I think are the goals of most people in this room. You know, how do we really fundamentally bring together the nature and the nurture, genetic work, and other people who do work in biology with people who do work at the forefront of environments and environmental factors. I think all of this work in the aftermath of the pandemic, on the heels of civic rights movements, really comes at such an important time in our field. So I hope for all of us, we just leave having a better sense of what some of the challenges are that our field face, but also we feel inspired to be able to dig in and really capitalize on all of the great work that's out there. We have resources within Purdue. I'll just say, you know, we have a number of faculty that have been hired. Shiro, I don't know where you are, but came from UW and is joining our faculty after a postdoc at Harvard. We're looking to bring in graduate students. And more than anything, I'm really here to identify new collaborations. I really believe deeply in team science. I think it takes a village for us to do the kind of work that we're doing. So if you think along those lines, come find me and let's chat. So what I want to do now is tell you a little bit more about, am I doing this wrong? Okay. Tell you a little bit more about the focus of my research group and what we're doing. So I think of it as we are identifying questions at the intersection of the who, the how, and when around depression prevention. So I've been focused on trying to identify ways to prevent depression throughout my entire career. With respect to the who, we do a lot of work in the GWAS space, trying to identify people at highest risk for depression using genetic and other markers of vulnerability. We also do a lot of work focused on mechanisms. So how is it that exposure to something like childhood adversity might get biologically embedded and shape long-term risk for depression and other adverse health outcomes? This is what most of the remainder of my talk is going to focus on, specifically these epigenetic signatures, which Lauren, for those of you who were here yesterday, talked about that as these chemical tags that get added to your DNA sequence that don't change the alphabet of your DNA, so to speak, but change the function of your genes. You'll also hear a lot of the work that we're doing around when. So in other words, the idea that it's not just that you're exposed to adversity or that you're exposed to other kinds of environmental factors, but it's when you're exposed and how that may play a role in shaping DNA methylation or other factors. And then I'm also always interested, and my group does a lot of trying to build novel infrastructure for scientific knowledge translation. So in other words, let's not just do ivory tower science, let's figure out how to get our work out into the field, to the community, and make a difference in improving the lives of people. So big picture in terms of orienting ourselves around the question of when. So a lot of my group does work on the question of, how is it that the timing of childhood adversity shapes risk for poor health outcomes like depression? So if you turn to the literature, you'll see that there's a number of different theoretical models that have been posited in this space. So one focuses on just a basic exposure model. And the idea here is that if you've been exposed to adversity, the idea is that perhaps you have an outsized risk of experiencing depression or another adverse health outcome compared to people who've never been exposed. There are also accumulation models or accumulation of risk models. And here I'm showing the most basic dose-response relationship, but essentially saying that the more adversity you have, the greater risk of an adverse health outcome. You can imagine that these associations can be nonlinear. You can think about adding multiple kinds of exposures, but here I'm just showing this for repeated exposure to the same event. There are also recency models, and the idea of a recency model is that it focuses on the time since the onset of exposure. So the idea here is that maybe your risk of an adverse health outcome is greatest in the time period shortly after you've been exposed, but that risk decreases over time. And then finally, the sensitive period model. So the idea here is maybe there are stages in the course of the lifespan when exposure to adversity or some other exposure has an outsized impact in shaping long-term health. So we study this and we try to understand which of these models makes the most sense by way of the data that we see, because it has implications for how we intervene. So if our data are consistent with an exposure model, that suggests that we can intervene at any time. And our goal should be to try to identify people who have these histories of exposure to adversity. If our data are more consistent with accumulation models, that suggests we want to intervene early before people are accruing multiple years of exposure. If our data are consistent with a recency model, it suggests that perhaps we want to intervene quickly, although it also maybe suggests that doing nothing is okay because people will, their risk will reduce over time. And if it's a sensitive period model, it suggests that we want to intervene during those periods in development or shortly before of maximal sensitivity. So in that way, I think about these high-risk periods, or excuse me, as these sensitive periods as being both high-risk periods when adversity or other adverse exposures may be differentially important, but also they might be high-reward periods or windows of opportunity when enriching interventions could also be more beneficial. So in my group, we're trying to really generate the research evidence that will allow us as scientists to make more precise recommendations for clinicians and policymakers about when to intervene, and do that on a high-resolution timescale. So in other words, trying to get more granular than just saying early, or possibly substantiate efforts focused on things like the first 1,000 days. So I want to tell you a little bit more about the work that we've been doing in this space. A lot of this has been led by Alex Lussier, who is a former postdoc in my group, now a collaborator, and on the faculty at Harvard Medical School and Mass General. So like other people in the field, we've done analyses showing that exposure to adversity has a measurable biological effect on the epigenome. So one of these projects has been analyzing data from the Avon longitudinal study of parents and children, birth cohort out of Bristol, England. And they have data, repeated measures data for methylation signatures in about 1,000 mother-child pairs that were collected as part of this subsample. And what Alex did was to analyze multiple kinds of adversities, things that happen to kids within their home, all the way up to environmental factors like neighborhood constructs, and look at exposure to adversity binned in different developmental time periods, and look at that in relation to DNA methylation on, at about 500,000 loci throughout the epigenome. And so the models that we ran, and I could tell you more about this, it's this approach called the Structured Life Course Modeling Approach, or SLCMA, allows us to understand which of these different theoretical models may best explain variation in CPG site level methylation at sites across the epigenome. So in this analysis, we found that 46 significant loci were distributed throughout the epigenome at an FDR corrected value. And when we dug deeper into these results, we found that the developmental timing of adversity seemed to matter most in shaping these methylation signatures at age 7. And specifically, of these 46 CPG sites, more than half were identified as being influenced by exposure to adversity in early childhood. So in other words, with this repeated measures data, we could model the association between the timing of adversity and methylation at each of these different loci. And we found the greatest differences in methylation among kids who were exposed during that particular time period. We also had baseline data, so we had data from birth that allowed us to look and understand if these effects actually occurred postnatally, and our results suggest that they did. And Alex and our team are now in the process of wrapping up meta-analyses where we've been working to replicate this finding in other data sets. Spoiler alert, in the methylation, or in the meta-analysis that we are writing up, we're finding more evidence for middle childhood as a potential sensitive period. What's also really interesting from the meta-analysis is we're really still not seeing very much by way of accumulation. So I take that as a sign that we need to be doing more to understand about the developmental timing of exposure. We've also been doing work longitudinally, trying to look at DNA methylation patterns across development. So we've looked at data from 15 to 17 in this cohort. These are really interesting results. So we look at the same 46 loci, and we find that the the magnitude of associations are generally decreasing over time, but the direction of effects is consistent. And very interestingly, we identify later in development new loci that we hadn't seen before that are coming up for the first time, but here again also speak to the importance of that early childhood period. We've also done, or in the process of wrapping up, or have recently completed other work in this space. So I mentioned this meta-analysis where we're analyzing data from other cohorts. We've also done work to look at mediation. So in other words, can we find that methylation is potentially a mechanism explaining how adversity leads to depression risk? We have a paper there that's now under review. Fascinating spoiler alert on this is that the methylation differences that we find related to adversity in predicting depression are reflecting both positive or protective effects and risk-conferring effects. So in other words, methylation is both protective in shaping or in preventing depression and also associated with some risk. So in other words, our epigenetic differences are creating both biological vulnerability and resilience. We also have a systematic review we published on how the timing and type of socioeconomic status indicator shapes methylation. The take home there is that these constructs aren't interchangeable. They have different impacts on methylation. And then we also have a more detailed empirical analysis looking at socioeconomic adversity of methylation at age 7 in ALSPAC. We also have an R01 grant where we're studying the role of positive life experiences on biology. It feels so refreshing, I have to say, to not just think about doom and gloom and all of this negative stuff, but to think about, like, what does social support do? What is growing up in a supportive neighborhood environment? Grandparent availability, what does that do to us biologically? So I have a fantastic team of collaborators that I've been working with for almost 10 years on this, and we're very excited to keep going with this work. And with that, I will stop and thank my outstanding lab members, sources of funding, and I'm look forward to the conversations that I hope to have throughout the meeting. So thank you very much.
Thank you. Fascinating work. Thank you. So we have time for a couple questions.
You're closer to the mic. Go quick.
I'm Michael Culbertson at the Center for Demography of Health and Aging here at UW-Madison. Could you give a brief overview of the SLCMA method?
Okay, brief. Structured life course modeling approach. It is allows you to balance theory with good empirical methods. You essentially start a priori by trying to understand or I have an idea of what you think may explain the relationship between your exposure and outcome. So for example, we always think about sensitive periods for the most part. There are other times where, with our SES work, for example, where we've looked at mobility models. So this idea that you are exposed to a family economic shock, but then parents get a job, and so you've got this mobility of coming in and out of these poverty circumstances. So essentially, you take all of these theories and then you encode them into variables that you then enter into a least angle regression. And then what you get from that is variables that allow you to determine how much of the variation in your outcome is explained by your different variables. The way that we analyze it is that we look both at the amount of variation explained by the first variable, but we also, for some analyses, look at combinations of things. And that's been really interesting because it's allowed us to know for some dimensions, for example, like depressive symptoms, that it's almost like a two-hit model. It's not just that being exposed in early childhood matters, but it's also accumulation that predicts later depressive symptoms. Those binary or looking in a more nuanced way at multiple theories gets really complicated in the high dimensional space, but is something that folks can consider. We have a couple, all of our papers are up on our lab website. There's a great chapter that we wrote that came out, I forget when. This is where the pandemic skews your sense of time. it was published in the last couple of years, but essentially it's really a how-to and clear walkthrough with code and very practical guidance for anyone who's interested in using it. And I think I'm clearly biased, but I think it's a nice method that allows us to identify in an unbiased way different effect estimates. And I'll also say that when we've compared the SLCMA approach to a traditional EWAS or environment-wide association study, we find far more signals than just a standard EWAS. Hope that was brief enough to maybe answer one more question.
Yeah, I'm Patrick Turley at USC. So one question I had was how are you, this is maybe a clarifying question more than anything, but how are you able to distinguish between your different models when you have only one measure of, one epigenetic measure? You know, I mean, I see you have longitudinal measures of you know, risk through childhood, but only one measure of epigenetics. And so it seems like it would be difficult to distinguish between some of the different models that you described.
So the models that we're describing are describing the relationship between the exposure and the outcome. So the exposure has repeated measures data, but our outcome is fixed. So in other words, we're finding for our age 7 methylation results that its exposure to adversity during 3 to 5 seems to be the time period where we're seeing more methylation differences at this time period. And so when we then have repeated outcome measures, we can then extend that and look to compare what those association results are with respect to these different theoretical models in predicting that outcome. So I think this is, I don't think it's not as straightforward to bring in any kind of longitudinal data, I think, at least in the context of the epigenetic space yet. But I think part of what we're trying to really do is just move beyond thinking exposed versus unexposed. And what I think our results show is that using SLCMA or other approaches where you are able to analyze these repeated measures data, the juice is worth the squeeze, so to speak. In other words, we do see meaningful differences that are often missed when we just ignore all of that variation and just look simply at exposed versus unexposed. So in that way, it's a kind of cautionary tale for all of us to be thinking about the potential downsides that come when we just look at binary, you know, presence, absence of a given exposure, rather than considering how that exposure unfolds over development.
Thank you.
Thank you.
Our next professor is Dr. Stephen Shaw from the Wharton School, who's going to be presenting work on biological age and its value to marketing theory and practice.
All right. Hi, everyone. I'm Steve. It's great to be here. And so I'm a postdoc in the business school at the University of Pennsylvania, and this is joint work with Remy Daviet, who's in the business school here at Wisconsin, and my advisor, Gidi Nave. We're all in the marketing department. So this is sort of probably towards the more applied end of the talks that you'll see at this conference. And I'll be talking about how epigenetic data, biological age, could be useful in the realm of consumer research or even more applied in the realm of marketing. So why should we care about aging or aging consumers? Well, the population is growing older, and we're undergoing a demographic shift where the first time in human history we'll have a much larger portion of old age individuals than ever before, right? So this is a growing market, and consumers don't want to die. Most of us don't want to die, right? And they're willing to engage in activities and spend money in order to increase their health span and longevity. So if we look at just the anti-aging product market, right, this is already a large market, and it's growing in size as well. So these tenets here are going to produce challenges in society, but also opportunity for researchers and businesses alike. The colors have gone strange on this slide, but many researchers studying aging or marketing firms would simply use chronological age, the time since birth, to understand aging, and that's a relatively simple metric. And aging itself is this multidimensional process, right? So if we think about other dimensions of age, you could think about the psychological dimension, sort of how you feel or feel age relative to your chronological age, or the physiological condition, biological age, which is what we're sort of advocating for in this work. And the key takeaway here is that at a given chronological age, say an individual is 40 years of age, there can be variation in these other dimensions. So you might feel older or younger than your chronological age, or you might physically be in better or worse condition, right? And we can take advantage of that heterogeneity to better understand the aging consumer. I think the previous presentations have done a great job priming what epigenetics is and what epigenetic clocks are, as well as what biological age is. So I'll sort of skip over here. But basically, there are a number of ways you can measure biological age. But epigenetic clocks seem to be sort of the gold standard for this. And essentially what they are is they're taking a series of DNA methylation tags, running them through some machine learning algorithms, and coming up with a, they train them generally on health outcomes, things like this. And you come up with an aggregate score of one's biological age, which is what we'll use in this study. I'm going to skip over this slide here, but the only thing I'll mention is what's important to know about epigenetic data is that it's malleable and it's dynamic. So it can be sped up or slowed down based on a variety of different factors throughout one's lifespan. So that provides, again, sort of opportunity for interventions. There are companies out there that will give consumers information about their biological age. So this is true age, true diagnostic. They're very similar to like a 23andMe or an Ancestry DNA type company. Consumers will send a blood sample in, and they'll get back a report about their biological aging and how they're doing relative to their cohort, relative to their chronological age, right? So this already exists. I think it's important for us to have conferences like this, now, and have these discussions now, because as these technologies develop, commercialization, will also come. And if we can have these conversations now, we can discuss the ethical implications of the raw data as well as the aggregated versions of the data like biological age, and put policies in place to potentially protect consumers and also to use these data ethically or use them or figure out when and why and where these data can be acceptably used. Another example of commercialization coming, you can even get an ancestry kit for your dog. Has anybody gotten one of these? No? Oh, for a cat, okay, there you go. Yeah, so you can get these things and you can learn about your pet's genetics. And there is even an epigenetic version of a dog age test, right? It's only $170 and you get epigenetic sequencing of your dog to figure out the age, right? So these technologies are coming down, the sequencing technologies are coming down in price, and commercialization is happening, and there'll be a wider range of applications in the future. So in this project, empirically, we have a pretty simple goal. We're, as far as I know, the first in a business school realm to be interested in biological age via epigenetics. And so we're simply trying to demonstrate the distinct value of biological age to consumer research over and above more traditional constructs of aging, like chronological age and like feel age, how you feel relative. And we do that using consumer activities and spending data from the health and retirement study. So the health and retirement study is a longitudinal panel data study housed at the University of Michigan. Go Blue, sorry Badger fans. We have access to 5 epigenetic clocks from this study. These are second generation clocks, sort of more modern clocks, and we use those as measures of biological age. And then we have dependent measures for a range of activities and spending items. These are things like how often do you watch TV or use a computer or visit your grandchildren, or how much money do you spend on recreational activities like vacations, or how much money do you spend on your utility bill, things like this. So we did a small categorization study first to distill down these activities and spending items into more robust categories, and we come up with 5 activities categories, things like activities that are out of home or more social, and 7 spending categories, things like giving, so charitable giving, or recreational spending on, like I said, vacations, things like that. The key metric that I'll show in our main result in 2 slides is an out-of-sample R-squared value. So we're trying to just look at the variance explained by these different dimensions of aging: chronological age, fetal age, and biological age. And that's as computed through a 5-fold cross-validation procedure. But before I get to that result, just looking at things kind of descriptively. This is a single item, so this is walking 20 minutes per day, how often you walk 20 minutes per day, with chronological age on the x-axis and one clock. This is the DNA Grimm age clock, which is our representation of biological age here on the y-axis. So I think that what I'd like you to take away here is if you were to do a cross-section at one age category, so 60 years of age or 70 years of age, there is quite a bit of heterogeneity in biological aging, maybe plus minus 10 years. So that looks sort of like the top end of that hypothetical graph that I started off with. And then the second point is, and I'll put a caveat here because all of the analysis I'll show here is correlational. I'm not making any causal claims, but we'd like to say that there's some meaningful aspect or some story that you can tell with these data. So the more blue the dot is, the more likely the individual is to say that they walk 20 minutes per day, right? And I think you can see that there are more blue dots below that AB line than above that AB line. So you could tell the story that individuals who are biologically younger than their chronological age are more likely to walk 20 minutes per day, right? And that sort of makes sense. So these are those out-of-sample R-squared values for the different dimensions of aging that I mentioned before across our set of activities and spending categories. And the blue portion of the bar, that's chronological age. The green portion is fetal age, psychological construct. And all I'll say on this slide is that you can see that aging has, these dimensions of aging have a differential effect across a variety of activities and spending categories. Some activities, at least in these old age individuals in the health and retirement study, there's not much of an effect, like gardening or household activities, but something like giving or physical, spending on giving or physical activities, aging seems to play a factor. I'll then now reveal these red bars, which is the variance explained by epigenetic clocks, our measure of biological aging. And so the key takeaway is, big surprise, biological age as measured through epigenetic clocks does increase R-squared across a range of consumer activities and spending patterns. So there's some interesting things that I'll point out here. If we look at sort of like physical activities, right? These dimensions of aging, chronological age, time-based measure, psychological age or fetal age, and biological age, a physiological measure, sort of explain an equal amount of variance in the overall impact of aging on these physical activities, whereas some other categories, like recreational spending, have a differential distribution of how these dimensions impact aging. And in the case of recreational spending, actually the portion of variance explained by biological aging is greater than these other dimensions, right? So again, going back to trying to sort of get some meaning out of these data, if we think about the recreational spending, right? Recreational spending is a category that would be highly relevant potentially to marketers or businesses. A lot of types of items in there could be useful. And so I mentioned before, one of those items is spending on vacations. So let's imagine we are interested in consumer spending on vacations, and we only had information about one's chronological age. This is what that data looks like. We've got segmented based on chronological age into groups of 5 years of chronological age, and then we have the amount that's being spent. It's sort of relatively flat from 50 to 60 or so, and then you have this bump in spending at 60 to 65 years of age, and then a decline, right? This kind of makes sense. This is around retirement age, right? So as individuals retire, they have, this is an American sample, as individuals retire, they have more time and the ability to spend money on vacations, and so they do so. And that's already a pretty interesting trend in terms of aging on spending on vacations. But now imagine we had access to biological age data, and we can split or segment our consumers based on whether they're biologically older or younger than their chronological age might suggest. That's what this graph is here, right? And this sort of adds a real nuance and a richness to this spending pattern on vacations. If we look at the red line at the bottom, these are individuals who are biologically older, right? We don't have that bump at retirement age anymore, whereas individuals who are biologically younger, we see an even larger bump there. So you can imagine if you're, let's say, a travel agency, and you now have access to biological age data, that's pretty interesting and useful to you to maybe target different groups or segment different groups of consumers or serve consumers different types of travel packages based on their biological age, right? So this paper is under review at the Journal of Marketing. We have a pretty rich discussion around the managerial and marketing implications of biological age. And we also talk about future research in consumer behavior as well as in the behavioral sciences in general. I think one of the most promising avenues for biological age is using it as a dependent variable for sort of retest type studies. You could think about like a randomized controlled trial for an anti-aging product. So I talked about the anti-aging market before, right? There's a lot of sort of scammy type of products in that market. I think we can all agree on that. But you could add value to firms and to consumers if you could test that with a real measure of physiological aging, right? And we already see some papers that are doing that with some like diabetes and obesity drugs like Ozempic and metformin, I think. But you could expand that to other types of products too. We also propose a theoretical framework for the aging consumer in this paper. If this is work that interests you at all, I'm happy to share the paper. Please send me an email or we can talk offline during the conference. So with that, I will thank my awesome collaborators. It's Remy in the middle there and Giddy on the right. And thank you so much for your time.
Thank you. So Anya Prince from Iowa. So putting together Sarah's talk and your talk, I have just one clarifying question, which is, what did you control for any other factors other than this, just in terms of like disability, right? You can't try if you have a lower biological age. It might be also because of health outcomes that limit your travel. But then on the more practical implementation side as well, if we have findings eventually that say that historical practices have impacted minority and disadvantaged populations, and they're going to have lower biological ages. The example you gave of a travel agent is kind of fun and exciting, and people can spend their money in positive ways. We also have, and you kind of hinted at this, some wellness products that might be more nefarious, less helpful. And so to what extent then might we be setting it up so that there's a huge push to people who are already disadvantaged to spend more money on a biological processes that, or like their biological age that might not actually help that because of other structural problems?
Sure. So the first part of your question was to do with adding different control variables. There is a set of analysis in the paper where we do that. Basically the proportion of variance is explained by any of these aging constructs is lower. It's less exciting to show in a talk like this, but it's still there, and it varies by the different categories how much these other factors reduce the variance, right? The second part, I hear, I guess, maybe some concern about the use of these data, right? That's, again, for me, if we can have these conversations now, hopefully we can use these data, and marketers in the future can use these data in safe and responsible ways, right? So you mentioned individuals with disability or individuals who had challenges during socialization or critical periods, right? I think if we can provide protections for these types of consumers, they can be better served for whatever means is beneficial to them, right? So an individual who is perhaps disabled or has a older biological age, like that sort of flat line in the sort of travel agency example, it doesn't necessarily mean that they don't want to travel, or maybe they're unable to travel or spend money on vacations in the ways that they want to. Maybe they're not being given the right opportunities, and firms can actually, by understanding their biological age, give them the right types of opportunities for them to enjoy a vacation that's suitable for their needs.
My question is there any, well, put it a different way, how much added value is knowing genetic indices of biological age? If I were a travel agent, maybe I should focus on people who do gardening or who walk a lot. Perhaps that's a better way and a less expensive way of doing effective marketing. So it's interesting that biological age correlates well with these activities, but as indices to be used practically, it seems to me that those activities have enough, explain enough variance that you don't need to spend money on genetic index.
Fair point. I think that's an open question, and that's where we're going with this. Really, is to understand how much. Yeah, I can't tell you the answer how much exactly the variance, but the reality is, a lot of these marketing exercises that are done, in the field, they're done at scale, right, with huge sample size. So the margins, these small percentages in variance, are significant. So even sort of any kind of competitive advantage you can get, you could imagine. For example, like Nielsen, a market research firm, they already have a Nielsen neuro department, right? And you could imagine, so they already have neuroscience data. They do EEG and some fMRI as well. You could imagine them having a department to do with social genomics, right, where they are the ones who create these insights and invest the money into genetic and epigenetic data, and then sell aggregated information about that to something like a travel agency.
I had a question. Fascinating work. Thank you. So I recently had my epigenetic clock calculated. I am somewhere between 12 and 55 biologically, depending on the clock.
Oh, wow.
So yeah, exactly, right? So I'm either on my way to an early grave or I'm going to be a centenarian. I liked, PhenoAge said I was around 29, and I really liked that. So I'm going to keep that in my head. But so clearly, these aren't ready for consumers yet. And there's a lot, at the individual level, there's a lot of error. How do you think about that? When will we know when they are ready? And how does True Diagnostics report their results to consumers? Because I would be very confused as a consumer to get that report back, or and then what am I to do? Should I work on my health more? Am I doing okay? So yeah, kind of circling that square with, we know these work very well at the population level, but how are we going to get when do we know that these are ready for consumers? And yeah, just what your thoughts on that?
Yeah, that's a very fair point. I think, so the point on True Diagnostic, I think they just use one clock in their report, so you wouldn't have that kind of, but the question still remains, and the reality is, even a firm like TrueAge Diagnostic, they want to provide value to the consumer. So they're biased in that sense. We as academics can actually study the relationship between these clocks and where each clock or how each clock might be useful in sort of different realms. In this case, we do have an analysis that looks at the individual clocks, and across these consumer activities and spending, the DNA Grimm age clock seems to be the one that adds the most value. So that's why I used that in some of the more descriptive stats that I showed there. When will they be ready? Yeah, when will we know? Yeah, I don't have the answer to that, but again, I think we as academics are the ones who are going to be studying that and finding that out. And one of the next steps in this project is actually collaborating with a few of these bioinformatic firms so that we can sample and resample sets of consumers who have their epigenetic data already collected, and we can study the relationship between these different dimensions of aging and the different clocks. Yeah.
So this is just a practical question that I was confused about as you were talking. So is the idea underlying your paper that the consumers would purchase an epigenetic clock, and then the companies that produce those clocks would then sell that information to marketing firms for direct marketing based on a person's epigenetic? Is that the world that you're envisioning?
I mean, I think that is probably what's going to happen. That's already happening, with some of the data from like 23andMe and Ancestry, right? These firms, part of their long-term business plan is to build up these large data sets of genetic data and then potentially be able to use that for consumer insights that they could sell. the market for these direct-to-consumer epigenetic tests is less mature than 23andMe and Ancestry, but I don't see why that couldn't happen. And again, I think our perspective is if we can have these conversations in the academic realm, we can better understand when those data are useful, but also sort of get ahead of the ethical and policy implications.
Yeah, yeah. I mean, the ethical stuff's important. I guess from a practical point of view, though, the question that I have, though.
That was quite a hand wave on the ethical stuff being important.
It's important. I'm just thinking about, like, so 23, like, people who are buying these consumer genetics tests is, I mean, it's something maybe like 5 to 10% of the population have purchased these, right? So it's a pretty small fraction.
There's still millions of them out there.
Sorry?
There are millions of consumers who've done it, though. I mean.
Yeah, but it's a small fraction of the population. A lot of these companies are really struggling now, right? Because it's like people get it. I think people buy them with the idea that I'm going to like learn about my ancestors. I don't know. I guess I would have to think about how with epigenetic stuff, because it matters like when it's taken too. You need people to repeatedly get these epigenetic measures, and for I think marketing companies to be able to use this kind of as people are aging, unless you think there's sort of persistence in epigenetic age. I guess that's just something I was thinking about as you're talking about, like in a practical sense, how big would this market have to be? Is that feasible given the size of the markets for genetic testing? Because I think epigenetic would be even harder than that, right?
Well, there's a couple things going on there. I think you're talking about scalability, right? And I think the scalability of these data types becomes more feasible as the cost of the sequencing technologies comes down, and we know that's coming down exponentially, right? And then I think your second point on testing and retesting, again, this is an open question. There's probably value from a single epigenetic test, but there's also probably more value from getting multiple tests over the lifespan. Obviously, getting multiple tests is going to be more expensive and more challenging. A single shot, like we're using single shot data here, right? And we think that this is, of value. So open questions, but fair points, all of them.
All right, thank you very much. Thank you, guys.
Okay, hey everyone, welcome back. We're getting ready for the next session.
Oh, yeah.
So this is going to be the third session for today's conference. It's going to be about families. And I want to say two different things about this session. First of all, I'm Chong Shi. Most people call me Q. I'm an associate professor in the biostats department here in Wisconsin. And I want to say two things about this session. First thing is, a lot of what we do in my research program is statistical methods for general human genetic research. But this family-based analysis is really one area where the community of human genetics has just beginning to learn from what social scientists think and why they care about this type of problem. And in human genetics in general, people have been doing GWAS in the last 20 years, and it gets really boring. It has become such a standard pipeline, just correlating millions of SNPs with some kind of outcome. Everybody is using the same kind of statistical framework. Then we begin to realize that parents' genome or family relatives' genome apparently have this major confounding effect. The simplest situation is the parental genome can have this indirect effect on children's outcome, and that actually surprised a lot of people in human genetics. And people in human genetics really just started to look into this on even non-social economic outcomes, such as neurodevelopment, cardiac development, and beginning to think about, for example, how maternal genotype can contribute to things like birth weight or neurodevelopmental disorders. So this is a lesson human genetic community really has learned from discussions we have in social genomics. And also, the second and third talks are going to be more about sort of mating. So in human genetics, it's taken for granted that in your data, you have Hardy-Weinberg equilibrium, there's random mating assumption, and people just pretend, we don't have to ever look at this again, but then you begin to get weird results. You get genetic correlation between height and cognition, and then people whenever see something like that in human genetics population, just pretend it's not there. We don't know how to interpret that, we don't discuss that. But then in social genomics, obviously, sort of mating is a tier one topic of interest. A lot of people are interested in that, and then social scientists are beginning to really incorporate genomic data into existing models that quantify sort of mating. And then this type of interdisciplinary work really has provided a lot of insights into the mechanisms behind some of the weird results we have seen for years in human genetics, and people are just beginning to develop new approaches to correct for this sort of issues in human genetics. So this is the first thing I wanted to say about the session. The second thing is very exciting to see that we have quite a bit of diversity in terms of career stage in this panel. Because the first presenter is a faculty member, but then the second and third presenters are both pre-doctoral trainees, which is really good to see. So, challenge them, ask questions, but also be nice to our academic babies.
Not to me.
Not to you. So, I guess we can move on to our first presenter, Dr. Leandro Carvalho, who's going to talk about, the topic of his talk is going to be a chip off the old block, genetics and intergenerational transmission of SES.
Thank you. So this is joint work with Marina Aguiar Palma, who's in the audience, Silvia Barcelos, and Shurvan Alton, and Tito Scalama. And we are acknowledging our funders who make this work possible. So as you of course know, the children of parents of higher SES are themselves more likely to achieve higher SES in adulthood. And for whatever reason, it seems that society is getting more and more concerned about this intergenerational transmission of SES. And we think that both genetics and environment may contribute to that, right? And in order to design effective policies to promote economic mobility, we have to understand how those two things jointly shape this intergenerational transmission. And social scientists have studied extensively the importance of environment, but we know much less how genetic contributes for this intergenerational transmission. Okay. So I'm sort of here just talking about field studies, but what I want to highlight is that for the longest time, we had to rely on twin designs or adoption designs to just overcome the fact that we didn't have genetic data to study this topic, and it's just more recently that we started using molecular genetic data and polygenic indexes to look at this question. The work by Dan Belsky and Jason and co-authors, and by Jonathan Lauren and co-authors are examples of this. And our approach is going to be somewhat similar to that. Okay, so what is the specific question that we are interested in is how does the genetics of one generation is going to affect the economic prospects of the next generation? And our genetic measure is going to be the workhorse, which is genomics, the EAPGI, the polygenic index for education attainment. In particular, we're using the EA4, but we don't have 23andMe. So we're going to be, so I think that we think that that hopefully the way that we can make progress with this question is by combining two things. One is going to be that we use a family design in order to be able to estimate the causal effects of the PGI of one generation on the socioeconomic status of the next generation. And the second thing is that, and for that, right, we're going to need genetic data from two generations. But in order to be able to sort of estimate the effect of this intergenerational transmission, we're actually going to need phenotypic data for a third generation. And the second element here is going to be merging the genetic data from these two generations with rich phenotypic data coming from administrative tax records in the Netherlands. Okay. And the other thing that we try to do in the paper as well is try to sort of start decomposing this sort of total effect by taking advantage of that every generation we have a Mendelian inheritance, so you can take advantage of that to actually be able to talk about mechanisms, and I'll get there. Okay, so we know that once we control for the genetics of one's parents, the variation that's left in one's genetics is random, and that applies to the PGI, and several studies have leveraged this variation to estimate direct genetic effects, so estimate the causal effects of one's PGI on one's own phenotypes, right? So, and here it's sort of the left-hand side, the dependent variable is some phenotype of this individual Y, and then on the right-hand side
you have this individual's eye PGI, and then you're controlling for the sum of the PGIs of this individual's parents, right? So several studies have used this approach. What we're going to be doing is something very similar. The big difference is that we're going to be changing the dependent variable, the left-hand side. So the right-hand side is going to remain exactly the same. The difference is that now on the left-hand side is going to be actually the phenotype of the individual eye's offspring, so not of the individual eye. Okay. So let me sort of give more details about that. So we start here with individual eye, and those are the parents of the individual eye, and then this individual gets married or not, but they do have to have children together. So this is going to be our 3 generations. We're going to be concentrating on just one side of the family tree. We're not going to look at the other one. And then I'll be referring to this sort of as 3 generations. So the first generation is going to be the oldest generation, if you want, the grandparents, and then the second generation, the parent, and then the third generation, the offspring, the children. Okay. So in order to, and that's what we're going to be talking about is what is the causal effect of individuals' eye PGI on the socioeconomic status of her offspring, or prefer what is the causal effect of the second generation's PGI on the socioeconomic status of the third generation. And to implement our empirical strategy, we're going to need the genetic data of individual eye, and then genetic data of individual's parents, and we're going to need the phenotypic data of the individual eye's offspring. Okay. This data is going to come from the Lifelines Biobank, the Dutch Lifelines Biobank. So individual eye necessarily participate in the study, and that's how we have their genetic data. And then for this individual's parents, in most of the cases, we're imputing their genotypes using Alex Young's method, because one or more siblings of individual eye actually also participated in the Lifelines Biobank. Or in fewer cases, there was one or more parents of individual eye who actually participated in the study. And then the data, the phenotypic data for individual's eye, that's coming from the administrative text records provided by CBS. So let me give you just a little bit more details about that. So we have the universe of taxpayers in the Netherlands. That's annual data from 2006 to 2021. And we have detailed data on income, so individual earnings, individual income, household income, and then wealth is decomposed, so net wealth is decomposed into debt and housing wealth and financial wealth and so on and so forth. But because we are interested in these outcomes in income and especially wealth, of course, individual needs a time in order to accumulate wealth. So what we're going to be imposing is that the individuals in the third generation, they need to be 30 years or more. Okay. So when we do that, we end up having a sample that in the second generation we have 5,700 people, and in the third generation we're going to have about almost 12,000 people. Okay. And because we have multiple years, when we look at that second generation, we're going to have about 80,000 observations. When we look at the third generation, we have about 100,000 observations. Okay. Okay, so that's actually our main results. So this table is showing the effects of the second generation's PGI on the third generation's socioeconomic status. Okay. So the rows here, they're showing the different phenotypes of the third generation. In the first 2 columns, the phenotypes are measured in levels. In the last 2 columns, the phenotypes are measured in percentile ranks. The odd columns show the point estimates, and the even columns show the standard errors. And the PGI of the second generation is measured in standard deviation units. So, and here the table is showing only the coefficient on the second generation PGIs. All of those are sort of controlling for the PGIs of the first generation, but I'm not showing that on the table. Okay. So, for example, if you look at the very first number there, the way to read that is that increasing the standard deviation, increasing the PGI of the second generation by 1 standard deviation would increase the number of years of schooling of the third generation by 0.3, or increase their likelihood of graduating from college by 7 percentage points. If you look at income, for example, it's saying that this 1 standard deviation increase in the PGI of the second generation would move the third generation by about 1.6, 1.7 percentiles in the distribution of income. Same if you're talking about distribution of net wealth. When you look at financial wealth, it seems that the effects are a little bit larger, more in terms of more around 2.1, 2.2 percentiles. Okay, so the takeaway for this table is that the PGI of one generation, or the genetics of one generation, does affect the income, the education, and the wealth of the next generation. It may be hard to sort of get a sense, are this big, are this small? One thing that we find very useful is actually compare that to the direct genetic effects. Okay, so the light blue bars there are showing the direct genetic effects, so it's showing the effects of the second generation's PGI on the second generation's own phenotypes. Okay, the phenotypes are showing the x-axis, and then the dark blue bars are showing the effect on the third generation. Right, so it's the same shock. Is the shock and the increase in the PGI of the second generation, but we are seeing how the effects of this shock across these two generations, right? And what I take from this figure is that there's a large persistence of these effects, right? So for most phenotypes, it's at least 2/3 that the effect on the third generation is at least 2/3 the effect on the second generation. So it's a very high persistence. And this is a good segue into talking about mechanisms. I've been talking about the sort of, let's say, total effect of the second generation's PGI and the third generation's SES. But there are two ways that this effect may take place, right? One is the second generation transmits part of their genes to the third generation. And then the genes of the second generation are just going to affect the income, the education, the wealth of the second generation, and consequently the environment in which the third generation grows, right? So you have those two things, and they jointly are going to shape the SES of the third generation. Okay, so we're going to try to sort of separate between those two channels. The right-hand side is the family background channel, and the right-hand side I'm going to refer as genetic transmission. I mean, I just want to make clear here that, like, even if we find that this right-hand side is sort of small, that doesn't mean that environment does not matter, right? We're just saying that the effect, the genetics of one generation on the other, how much that goes through environment and how much of that goes just through the genetic transmission. It doesn't say that environment does not matter. Just be careful about that. Okay, so that's the genetic transmission channel. So we know the total effect. So if we can just quantify one side, we get the other for free. So what we're going to do is actually try to quantify the genetic transmission channel because we know the red arrow, right? If there's a shock to the second generation, about half of it should be transmitted to the third generation. I should say that I'm like, just for saving for the next speaker, I'm not going to say anything about sorted mating, so I'm making kind of simpler here. And then you would need to know that the blue arrow, right, which is it's just really the direct genetic effects for the third generation, right? That's really what it is. So so that's a way that we could possibly estimate the sort of left-hand side. It turns out that we've tried to use a single sample to estimate both the total effect and this sort of direct genetic effect for the third generation. We end up with a very small sample. So what we end up doing is estimating those two effects in two different samples and trying to make the two samples as similar as possible. So the ones that we used to estimate the total effects, they were born between 64 and 91. So we impose that same thing for the ones in which we are estimating the direct genetic effects and reweight that sample to make them look as much as possible as the other sample in terms of gender and year of birth. Okay. So, and here's what we find. So the first two columns, they are just reproducing the results that I showed you before. The effects on income and wealth here are all in percentile ranks. I'm not showing you anything in levels. Then the genetic transmission was this left-hand side of the figure that I just showed you. And the very last two columns is just a ratio, calculating dividing the genetic transmission by the total effect. And so if you look at the sort of before to last column, because that's the really important thing, what we take from that is that when it comes to education and income, it seems that the genetic transmission would explain about 2/3 or even more of the effects. So the family background would play a sort of smaller role. But when it comes to wealth, it seems that then that family background channel seems to play a bigger role, which we actually find intuitive because there are sort of some really funky laws in the Netherlands that allows parents that are sort of higher SES to sort of give a lag to their children to actually buy their first homes. But I won't have time to get into details. So that's consistent with that. Oh, sorry, I'm going backwards. Okay, so we merged molecular genetic data with this very rich demonstrative text data from the Netherlands, and we use the family design to be able to isolate causal genetic effects, and we find that genetics of one generation does influence the education, income, and wealth of the next generation, so it does seem to contribute to the intergenerational transmission that we see in the data. And when we try to sort of look at those two mechanisms that we talked about, when it comes to income and education, it seems mostly really through genetic transmission, but when it comes to wealth, the family background plays a bigger role. Thank you.
Time for some questions.
Great presentation. So my question was, in terms of when you look at the effects on education attainment, is it conditional on someone finishing high school, or is it open to if someone drops out like from early on, that gets included, or?
I mean, we're including all educational levels, yes.
So my thinking was like, would the effect be sort of a lower bound? Because if someone drops out and does not get included early on, so would that indicate the effect sizes would be a lower bound? And the last question
included is where? Sorry,
so in your data, like everyone gets included, like like a student like maybe dropping out from elementary school, like, would that end up in your sample?
I mean, I don't know if it could affect volunteer bias. It may be that people there are less educated, are less likely to agree to participate in the study. The recruitment was done through GPs, for general practitioners. But it could be. I mean, if there is some kind of, if people with lower education are less likely to go to the doctor, it's possible that it could influence. Yeah.
Because the PGI of someone dropping out like really early on would be really relatively lower potentially, so that might affect the estimates. And the last one is like whether you were able to get data in terms of educational achievement. So performance on a test score would give you probably more insight into the effects of.
Yeah, so we have used sort of the basic data that they have in CBS on education. I know that there's sort of a test that kids take earlier, but we haven't gained access to that yet. So maybe if we do, we may be able to see things. But the thing is like you see that the third generation, we don't see them in the Lifelines biobank. So all the data that we have about them is coming from the administrative data from the CBS. So they don't have as many sort of rich data on education. Thank you.
Thank you. I was curious whether you looked within families at the second generation to see if the effects persist, because you have siblings at the second generation level.
Mm-hmm. So I don't know if that's exactly what you're asking, but we did look at like, does the effect of the second generation PGI affects the phenotypes of the siblings of these individuals in the second generation. So it's kind of like horizontal and not, and we don't find any effects. And we don't find effects also in the nephews and nieces. So if I look at actually the cousins of the offspring, we also don't find any effects. Yeah.
Okay, interesting. Thank you.
Thank you.
Another question?
Yeah, super interesting work and great presentation. Made it really easy to follow, so thank you. I guess just kind of your thoughts at this stage point on what the implications are for policy here. I know that's a loaded question, but since you queued it up that way, and what you think this might look like in the United States if we did have the data to do this in the U.S. Do you think inequality would play a larger role where we might see more, genetic transmission of, rather than more genetic transmission, family background playing more of a role in some of these outcomes? Or yeah, just curious to hear some of your thoughts on that at this stage. Thank you.
So in terms of policy implications, I tend to see most of this as basic research and be very careful about saying anything about policy. And going back to a point that you made earlier, also when it comes to genetics, we know that predictions at the individual level are sort of, it's not good enough, it's just at the population level. But I guess if you really push me, what do you think we can learn? I guess what we can learn is, for example, if you were more worried about inequality in wealth, right, then you see that things that go to the family background, so any interventions that have to do with the family environment, that may help to reduce this inequality, right? If it's other things like income and education where you see that most is genetic transmission, then you have to find ways of interventions that would affect the direct genetic effects for this third generation, basically. Like, how do we make sort of, right, these genetic effects that we know they're not just biologically immutable, they are really sort of a function of the environment. Can you change the environment to actually change the direct genetic effects? So I think that's it. Oh, and I mean, the US, my hunch that the effects would be bigger.
Yeah, interesting. All of us.
Yeah, thank you very much. We should move on to the next presentation. Our next speaker is Qingwen Zheng, who is a, that's correct, right? The next talk is, I believe, Qingwen Zheng, actually, who is an undergraduate student at UW-Madison, and he's going to tell you about genetic basis of partner choice.
Okay, so hi everyone. My name is Qingwen Zheng. I'm an undergraduate student at UW-Madison. I'm right now working with Chongshi on statistical genetic research. So today I will talk about the genetic basis of partner choice. I will discuss how the genetic drives our human partner choice, human marriage partner choice selection, and also I will talk about how do we use this kind of knowledge. So this work is guided by Chongshi and collaborated with Sjoerd and Turkio from Netherlands and Norway. So many of you guys here should know assorted mating. So partner choice is now random. People choose their spouse that are relatively similar to themselves. In social science study, assorted mating received broad attention. You can use the information study from assorted mating to have many explanations to societal phenomena. And in genetics, assorted mating will increase phenotypic variance, and also it will induce correlation between two independent variants in your chromosome. So it will definitely bias many genetic analysis, including genetic correlation analysis, heritability estimation, and if you run a standard GWAS, the results will also be biased too. And also, many existing literature also show genomic evidence that some trait is under assortment. You could either use your own PGS to predict your spouse phenotype, or just test the correlation between polygenic risk score between odd and even chromosome to show whether a trait is under assortment for past generations. However, this often result is under assumption that trait-specific sorting, or just show results that some certain specific trait is under assortment. It is definitely an oversimplified model for assorted mating. As we all know, partner choice selection must involve ton of characteristics of individuals, and you should involve many trade-offs between all the characteristics in the model. So how could we do this? How do we have a better framework to quantify the authority mating? So it turns out to be some well-defined model in economics. And we built this model with some collaborators. So everyone in the partner choice market have an index, and any partner preference is based on this index in the partner market. So as I just mentioned about, this process should involve tons of characteristics and trade-off things between characteristics. So we can consider this index as a summarization of high-dimensional data. But there's no explicit function or algorithm to tell you how do we compute or how do we get this index. So basically, in general, authority mating is a couple resemblance. But how do we quantify this? We can't just use the within couple correlation for traits like education or height to quantify the couple resemblance. It's this oversimplified assumption, as I just said. So here we use the index to quantify the couple resemblance, to quantify authority mating. And we said that people choose their spouse with similar index. So our optimal goal here is quite intuitive. We want to run a GWAS on this index, and through that we can get some genetic insight of how the genetic drives our partner choice selection. So they will be useful we can use this information, use this GWAS to do some correction to existing post-GWAS analysis. So here I'm talking about genetic correlation analysis. But how do we implement this? As I'm talking about to do a GWAS on an unknowing variable, and we even don't know how to quantify this. We think the answer is buried in spousal outcomes. It says that your spouse characteristics suggest some of your own index information. I will use an example to show this. And so here we just assume height information positively influence the index information. So overall, taller people have a higher index. This is just an assumption. So knowing that your spouse is a tall person, what that tells you, the first information come to my mind, I might also be tall because we're all familiar with this kind of trait-specific assortment. But if we talk about the index model I just mentioned, so all the characteristics of your own contribute to your index. So to have a similar index with your spouse, I could not, instead of being a tall person, I could also be a well-educated person or a richer person if we assume education and income positively influence the index. And if we take one more step further, think about how the information that my spouse is a tall person influence my index information. So basically, as we assume, height positively influence the index. So conditioning on this information, the expectation of your spouse index will be higher. And as the similarity between my index and my spouse index, I will have a high expectation of my index. So overall, in general, we can say knowing my spouse is a tall person essentially tells you I may have a high index. And most importantly, we can switch the height information to many other characteristics. For example, here I use the income as an example. I can tell the exactly same story as I just mentioned. So basically here is that we can conclude that spouse information tells some level of my own index information, and different spousal outcome suggest the same thing. They are similar informative on own index information. And also we have a really nice mass derivation to show this result. So under some necessary assumption, we show that if you do a GWAS on spousal outcome, it is essentially a GWAS on partner choice index up to some scaling factor difference. And also this equation suggests the different spousal traits are similar informative on your spouse, on your own index, which is a key intuition in our study. One example will be, well, BMI and income are quite different characteristics, but they are both similar informative on partners' value in the partner market. So of course, it will be really boring to show all the math detail of how we get here, but I will show some real data evidence. So first is that we show how we collect our sample. We collect sample from 4 major biobanks, UK Biobank, Lifelines, MOBA, and HRS. Overall, we get over 100,000 non-Hispanic white spousal pairs, allowing us to do the largest ever spousal outcome GWAS. And we did it for 4 phenotypes: spousal BMI, income, height, and education. The first evidence is that we observe a significant heritability for spousal trait GWAS, suggesting your own genotype is explaining your spouse genotype, your spouse phenotype, sorry. And And also this significant heritability estimate allows us to do more downstream analysis to the spousal trait GWAS. And the second and the strongest evidence to support our model is that we observe a substantial higher genetic correlation among spousal trait, which is the upper triangle of panel B, than the genetic correlation among own trait GWAS. For example, you can see spousal BMI and income are near perfect genetic correlation of -0.9, but own BMI and own income are quite different traits. Then we have genetic correlation around -0.3, which is strongly support that statement I just made. Different spousal traits are similarly informative on index information. However, we also notice that the estimates on the upper triangle, the genetic correlation between spousal traits are not 1, suggests that instead of the index, partner choice index, spousal trait GWAS also captures something else. One thing we can think of is that trait-specific sorting. Example will be well-edited person choose higher EA person only because they have higher access to them. It's not because the index sorting framework works here. So intuitively, if we can get rid of this trait-specific noise and extract the shared component of different spousal trait GWAS, we can get what's left to us is the index GWAS. So to do this, we use Genomic SEM. The software allows us to extract the shared component of different GWAS. And we use the spousal trait GWAS as input. We obtain a great model fit. And here is the Manhattan plot for this powerful index GWAS. So after getting that, we run many genetic correlation analysis with our index GWAS. And among all the 60 traits we include in our analysis, we found education has the highest genetic correlation with the index, suggesting higher education genetically correlated with a higher index. And trait and height at BMI are also significantly genetically correlated with index, but with different sign. And we found many smoking behavior is genetically correlated with lower index, but drinking alcohol behavior is not correlated with the index. Also, we found many health outcomes have the negative correlation with the index. But we know is that education also has the negative correlation with many health outcomes, and this index GWAS is highly correlated with education. So we are thinking like whether the education drives the negative correlation between health outcomes and this index. And it will be insightful to see the part of the correlation that not explaining by education alone. So to do this, we use software from our group, GSUB, which is an analytical approach for GWAS by subtraction. For what it does is basically it can decompose the GWAS to two orthogonal components. So here we decompose index to EA and a non-EA component, and use the non-EA component to run the genetic correlations, and the result will be like the part of genetic correlation of index that's not explaining by education. So basically, we found that for some trait like BMI and height, the significant remains, but shows moderate reduction. But for some reproductive trait and the smoking trait, the significant also remains, but showing a substantial decrease. The most important thing here is that we found after removing the education component of index GWAS, we found that genetic correlation between many health outcomes, including heart failure, LDL, neuroticism, tend to non-result, suggested that all of this negative genetic correlation we just showed in the previous slide is driven by EA alone. So after that, we want to say, is there a difference between male factor and male index and female index? I mean, the genetic component, whether the genetic component is different between male index and female index. So we first run a sex-stratified GWAS to spousal trait, and we found some results as previously reported in many social science literatures. First, the thing you should notice here is that the differential heritability in BMI suggests that if BMI values more to female index compared with male. Also, we did a sex-stratified index GWAS, and we found that a perfect genetic correlation, near perfect genetic correlation between male and female index. So the takeaway here is that, well, there is some difference between male and female index. Some traits loading is different to the index between male and female, but the overall index for male and female are pretty similar. They share a highly shared genetic component across genders. The last thing I want to talk about here is how do we use this kind of GWAS to do some correction to genetic correlation analysis. So our intuition here is that if you run a standard GWAS, the result will be biased from assorted mating. And if we can regress out the component that's biased by assorted mating, we can and use that to run genetic correlation analysis. Maybe you can have a better estimate. So we use the GWAS by subtraction again. For each population, for each standard GWAS, we get a non-index component for that GWAS, and we name it conditional GWAS, and use that to run genetic correlation analysis. Maybe could give us the results that reduce the bias induced by assorted mating. So for each genetic correlation between trait A and B, we have 3 sets of genetic correlation analysis. So the first is using standard GWAS as input, and the second is using sibling GWAS as input, which you can think as unbiased result. And the last one is using conditional GWAS, the GWAS by subtraction output I just mentioned as input to run genetic correlation analysis. So for genetic correlation between height and education, we found for population approach, we found a positive significant genetic correlation. But after factor conditioning, after this indexed conditioning, we found a null result, which is consistent to sibling-based result, and which is suggests that the genetic correlation between height and EA using population approach is raised by assorted mating. And also, if we look at the result, genetic correlation between cognition and education, we found that after the conditioning, the result is more close to population approach instead of sibling approach. We think it makes perfect sense, as we all know that cognition and education contain a large portion of indirect effect, and doing this kind of assorted mating correction won't solve the bias induced by indirect effect. So in conclusion, we first ever did a GWAS on marriage partner index, and we also did the largest ever GWAS on spousal outcome. Doing this allows us to have insight of how genetics drive our marriage partner selection, and also we can use this kind of knowledge to do some correction to existing post-GWAS analysis. So I will thank to all the co-authors for this paper, and the 3 biostatisticians in our lab helped this project a lot. Of course, the UW Medicine Social Genomics group for providing a lot of helpful comment to this. And in the last, there will be like a self-advertisement. So I will apply PhD many programs for the next application season. So thanks, guys.
Probably this is a dumb question from the sociologist, but I think you are assuming the different sex marriage, but probably you are excluding same-sex marriage because you are saying in female and female. Would you mind reminding me the proportion of the same-sex marriage, and how did you deal with the same-sex marriage and this analysis?
Yes, we exclude the same-sex partner, but I'm not very sure about the portion of how much a same-sex partner is included in each biobank. Maybe we can discuss this, and maybe it's possible to put it in the manuscript.
So part of the reason, part of the reason you motivated this talk was because assortative mating can affect GWAS. Mm-hmm. And so you're doing a GWAS of assortative mating, but you're using population data. And I was wondering to the degree to which assortative mating may be affecting your results. Like, are we doing something circular if assortative mating can affect a GWAS of assortative mating?
Yeah, we have tried a sibling-based approach, but if you want to run sibling-based GWAS for the spousal outcome, you need to have a spousal pair and two a sibling pair, and two siblings need to have a spouse information in the biobank. So after the filtering, we found there's only 600 sibling pair in UK biobank. We couldn't use this GWAS to run a powerful GWAS. And also we compared the regression coefficient estimated in sibling setting with our population setting. We found that it's quite a consistent result. There's no much flip sign things happening.
You could do it I agree empirically, it wasn't feasible, but you could simulate it and see kind of what the effect of what would be the effect in a large simulation, right?
Yeah, but we haven't included any simulations in this project, and maybe we can consider this in the future.
Well, being too nice to the meeting. If there are no more questions, we can move on to the third presentation. Our third speaker is Matthew Hao, who is a post-bac researcher at UCLA. He's going to talk about assorted mating across time.
Awesome. Where's the clicker?
Oh, sorry.
Oh. Yeah, I thought it was going crazy. All right. Hi, everyone. My name is Matthew. I am a pre-doc researcher at UCLA working for SSGAC. And today I'm going to be talking about assortative mating across time, or as we like to call it, a MOT. And I was mentored under Patrick Turley. So, first of all, I wanted to talk about what exactly assortative mating is. I think my predecessor did a good job, but just in case we forgot, assortative mating is a pattern in which individuals with similar phenotypes or genotypes mate with each other more frequently than what we would expect under random mating. And so Amat asked the question, how does the degree of assortative mating change across time? And we might be asking the question, why are we studying assortative mating? Well, first of all, if there's assortative mating on disease phenotypes, we can imagine that assortative mating can affect disease prevalence and severity. And then on top of that, assortative mating can actually reflect social phenomena, which is probably interesting to a lot of people who do social science here. We can think about inequality, social mobility, and so on. And an example phenotype that we can think of here is assortative mating on wealth. So now let's talk about how we're going to measure assortative mating. To do so, we're going to use polygenic indices, or PGIs, which are just weighted sums of alleles. Now, if we consider parents M and F having children at time t, then we can define their PGIs as PM and PF. And then we're going to define assortative mating as the correlation between PM and PF at time t. Now, it's more of an ideal to have data on these parental PGIs. And so what we're going to do instead with Amat is we're going to approximate this correlation using the PGIs of their children born at time t. And a key feature of assortative mating that we should talk about is that it induces both within and cross-chromosome linkage disequilibrium, or LD. And what Amat exploits here is that cross-chromosome allele pairs vastly outnumber within chromosome pairs, and we can get a very close approximation of the degree of assortative mating by just focusing on the contribution of cross-chromosome pairs to the total PGI variance. We're going to do so using the PGIs of the children. So without proof, let's build an approximation of our measure of assortative mating, MT. First of all, I just wanted to define X. X is just going to be the set of all cross-chromosome allele pairs. And now let's talk about the numerator of our approximation. We're going to define Vx of t as the contribution of all cross-chromosome allele pairs to the whole genome PGI variance. And so what this is doing is it's kind of a raw estimate of assortative mating. It's a raw measure of assortative mating that takes into account the linkage disequilibrium, the cross-chromosome linkage disequilibrium. However, this measure of assortative mating also includes assortative mating that might have happened in previous generations. And so this second term in the numerator here is subtracting out that assortative mating that may happen or may have happened in previous generations. Now, at the end of the day, we are approximating a correlation coefficient, And so this term in the denominator here is just normalizing. And then also, as you recall, we are using cross-chromosome allele pairs to approximate assortative mating. But as we talked about, assortative mating induces both within and cross-chromosome linkage disequilibrium. And so we include an inflation parameter, pi, which just scales our approximation of assortative mating to account for the fact that assortative mating also induces that within chromosome linkage disequilibrium. So now let's talk about the data that we're using to produce our results, and I'll show you that in a second. Our results are based on tranche 3 of the UK Biobank for individuals with European genetic ancestry, and we're using the largest GWAS summary statistics available to us. Now we're going to use 2 different types of PGIs to approximate assortative mating. First of all, we're going to use within-family PGIs. These are based on currently unpublished data. An important note to make here is that the GWAS has lower power just due to having lower sample size, but the PGI is a noisy estimate of the additive SNP factor. The second type of PGI that we're going to use is based on population GWAS. Just due to higher sample size, like I was saying, this GWAS will have higher power, but the interpretation of the PGI is much less straightforward. Another note about results that we should talk about is that AMOT will give unbiased estimates of the correlation of parental PGIs, which is great, and that is regardless of the PGI type, so if we're using population or within family PGIs. But it's important to note that population within family results will have different interpretations. Our within family PGIs are a noisy estimate of the additive SNP factor. So from our AMOT results, we can infer the correlation of the parental phenotypes. However, also we should notice that because there's more noise in the PGIs, there may be an attenuation of the assortative mating estimates that we're going to look at. On the other hand, the interpretation of the population PGIs, we can't infer the correlation of the phenotypes since these PGIs are biased from the AMOT results. So that's something important. And also, let's talk about one of our methods that we're using. We're going to residualize the PGIs on the principal components of the genetic data. And we're going to do that to adjust, at least partially, for population stratification-induced assortative mating. We can think of population stratification as a form of assortative mating, actually. And a simplification we can think about here is that population stratification is just assortative mating on geography. And so let's actually look at a plot generated by AMOT. So I'm a bit of a walker, so I'm going to walk if that's okay. Just let me know if you can't hear me. So on the y-axis of this plot here, we can see our measure of assortative mating, M of t. And on the x-axis, we have the year of birth of the people that we have PGIs for. And as we can see, assortative mating on latitude, this is latitude, by the way, I should have explained that before. So this is an assortative mating plot for latitude, which is geography. So between 1954 and about 1970, we can see that we have a pretty large signal for assortative mating on latitude, up to about 0.4, 0.5. However, when we residualize on the first 20 principal components of the genetic data, we can see that that signal entirely disappears. So we can see that residualizing our PGIs on the principal components does a pretty good job of removing population stratification-induced assortative mating. And so all subsequent plots that I'm going to show you are based on principal component adjusted PGIs. So now let's talk about some exciting results. Something that a lot of people are probably interested in is education attainment. And I'm going to talk about the population PGIs first. So I'll explain a little bit about this plot. As we can see, our measure of assortative mating is statistically different from 0 for all birth years. And we can also see a pretty strong increasing trend in assortative mating all throughout the birth years. And to quantify this increase in assortative mating, we decided to add a linear regression. I hope you guys can see that. It's pretty small, but it's a little dotted line on this plot. And In the bottom left corner, we can see our slope and intercept estimates and the associated standard errors. And if we compare the standard error of our slope estimate to our slope estimate, then we can see that our slope is significant at the 0.05 level. So we see a significant increase in assortative mating on education attainment between about 1940 and 1970, which is really interesting. Now let's look at the within family PGIs. When we use the within family PGIs, we can see that those signals that we were seeing in the previous plot for population PGIs almost entirely disappear. We do see an upward trend. However, we see that assortative mating is not statistically different from 0 for any birth year in the sample that we consider. And on top of that, our slope is no longer significant. And also, just to note, these two plots are not scaled exactly the same. If you do, they look really funny. But just so you know, I did not scale these plots exactly the same, just that you are not deceived. All right, so let's talk about height now. Height is another interesting phenotype. And again, we can see that for all birth years, assortative mating for height is statistically different from 0. And on top of that, we again have a significant slope. So assortative mating is increasing over this period for height. If we look at the within family PGIs, we can see, again, just like education, that signal disappears. For almost all birth years, except for maybe around 1960, assortative mating is not statistically different from 0. And our slope is very clearly not significant. One last phenotype that I chose that we can look at today is ever smoker. As we can see, again, for population PGIs, we don't have a significant slope, but we do have a significant intercept. And we can see that we also have a significant intercept for our within family PGIs. So in neither case here do we have a significant slope, but we do have a significant result for within family PGIs that is interesting to look at. And here's just a summary table in case you guys were interested in any other phenotypes that we looked at. We have ages first birth, BMI, neuroticism, self-rated health, subjective well-being, etc. If you guys want to choose your favorite phenotype, if you're interested in any specific phenotype, I do have plots for all of these and some supplementary slides. I have both plots that just describe the differences between population and within family PGIs, and also I can show you guys the residualization process for all these phenotypes for both population and within family. But yeah, so as you can see, the only significant intercept that we have for within family PGIs is for ever smoker, which is what we looked at, and we have quite a few significant results for population PGIs. Now let's look at the slope estimates. Again, we can see that there is only one significant result for within family, and that is for cigarettes per day. Interestingly, we can see a decreasing trend here. If you guys are interested in that, we can talk about that some more. I can show you guys the plot. And for population PGIs, again, we have quite a few significant results, two of which I showed you. Another significant slope is for age at first birth. So conclusion is that our key findings, we have found evidence of recent assortative mating on a variety of traits, which is really interesting. And assortative mating appears to be changing over time for several traits. And future directions for this project include expanding the set of traits that we consider. And I'm happy to take some suggestions if you guys are interested. And also using the same framework to estimate cross-trait assortative mating. And I also, that got cut off a little bit. I also wanted to thank quite a few people, including Patrick Turley, who mentored me on this project, and also Dan Benjamin and Alex Young for providing feedback on this presentation. And yeah, that's all. Thank you so much. Oh, hi.
So one of the populations you're using is UK, am I correct? Is
Yes.
there any index of how about any subpopulation there that has family arranged marriages?
I'm not exactly sure, to be honest.
I mean, the question would be if that's a significant component, or if that type of assortative mating may generate differences in your data.
Right, that's very interesting. Yeah, thank you.
And this data is all European ancestry data, so presumably there's not very many European ancestry populations in the UK that have arranged marriages.
These are all European.
These are all European ancestry.
Got it. Thank you.
Really long wind up for the question. So given that, thanks for the presentation. Given that you're thinking about context over, or time, right? You have birth cohorts from 1930s to 1960s.
Right.
I'm curious how you think about changes in social context. So
Yeah.
income of women, I mean, especially for women, right? Income of women, age at first birth. There wasn't the same birth control for women who were reproductive in the 1930s, born in the 1930s as 60s. And so how do you think about that in terms of picking mates with these traits when the traits looked very different given the social context at the time?
That's a great question. Yeah, so our measure of assortative mating really is just kind of like a raw, measure of assortative mating that doesn't really take that into account. That's really interesting, and I'd be excited to kind of like look into that and see how that kind of like affects like how people are choosing their mates. Yeah, but I can't really say anything about that. But yeah, that's a very interesting point. Yeah, thank you.
Taking my moderator privilege. So sometimes you see this type of results in the literature that people will report differential predictability of polygenic score over time. So you see changing R square across birth cohort.
Yeah.
I wonder if we should think of assortative mating as part of the mechanism that has led to those type of results when we interpret those, or do you think that tells you something about the scores being differentially informative, and when you calculate this correlation, you should keep that in mind when you interpret assortative mating results?
Yeah, that's a great point. So especially for cigarettes per day, I can show you that. Let me see. Is that not working anymore? Oh, here we go. There we go. Thank you. I don't know if someone did something. I might have to skip through quite a few slides because I can't click the links. But if we look at our result, oh, there we go. For cigarettes per day, we can see that we have a actual significant decrease in assortative mating over time. So I guess we can call that like disassortative mating, perhaps. However, an important thing to note here is that mortality selection and also declining predictive power of the predictive ability of the PGI over time, simply due to people smoking less, can show disassortative mating. So that's definitely something that's definitely a concern to discuss when looking at these results. Yeah. Go ahead.
Great presentation. So I was just wondering, besides like assortative mating changing over time, it would be nice to maybe find a way to actually map it and like see, for instance, in the UK, like how that looks like. Like, do we see like assortative mating changing more in certain parts of the UK versus other? So that may give you also idea of like potential differences in societal values, culture, and things like that.
Right.
And it would be really interesting to see these results also like in the US and in other places as well.
Yeah, that would be amazing. Yeah, that would be awesome. Thank you.
Oh,
right, awesome.
One more question. You're the last one standing. You think as a
So I have, I guess, two questions in one. First, this data, did it come from census data that was there over time? Like, where did this come from?
Yep.
And then two, why did you use just latitude, and why not both latitude and longitude, or just longitude, considering that Britain is longer than it is wide?
Good question. So your first question was where does this data come from? So this data comes from UK Biobank, and we have access to both genotypes and phenotypes of every individual's right through UK Biobank through data buckets.
So it's year of birth. That's okay. It's not year of the event.
Right.
So it's not smoking. Oh, okay.
Yeah, exactly. Yes. Yeah. And then on top of that, I should also note that when running a mod, you do need to assume a distribution of generations, or distribution of generation lengths. And so we do get that information from the UK census. So that's an important thing to note, but not for the phenotypes themselves. And your second question, can you remind me what that was?
Why did you just use latitude, and why didn't you use latitude and longitude? And why wasn't longitude used instead, considering that Great Britain is longer than it is wide?
That's a great question. Yeah, so to be honest, there had just been previous work done on a mod, and latitude was the phenotype that was given in previous work. I don't know if we have access to a longitude phenotype through the UK.
And we do, but
we do.
Latitude is north-south, right? So that would be, if you want the thing that has the most variation, you would do latitude, right?
Latitude goes this way, and then longitude goes this way.
Yeah, but a person's latitude says how far north they are.
Yes. So you're going to have a longer span per that. If you're doing latitudes, those are smaller lengths, so you have less span.
I think ultimately it was mostly just illustrative to show that PCs remove all of this geographic stuff. But I think we could do both.
Okay. Yeah. Thank you for the suggestion. Yeah.
All right. Thank you very much.
Awesome.
Okay, so I'll just say a few words in addition before we launch into our talks today. I noticed that a lot of our presentations have focused on leveraging big data, existing data sets, and you're going to hear, I think, a lot about that today in our application session on existing data sets that are taking information that other people have collected and ways in which we can utilize genomic information and those other pieces of information that people have collected to try to better understand or predict how something happens, a phenomenology. But what I think is also very important to think about as we consider our talks upcoming is how have people been measuring the constructs that we are studying? I think this is particularly true when we start to think about constructs that are latent. I know one of the speakers had presented on a construct like depression. This isn't something that's as easily measurable as something like height. I do think that I hope today in our session to come that you're going to hear not only about the ways in which we've applied genetic information and environmental information to study these outcomes, but also a consideration for the importance of measurement and accounting for measurement error as well. So with that said, I'm going to introduce our first speaker, which is not Sally Kuo. Unfortunately, there was a travel snafu, but Jessica Salvatore, who's from Rutgers University, and she will be discussing a project called The Influence of Spousal Genotype on Alcohol Problems in Marriage. Take it away, Jessica.
Thank you, James, for that kind introduction, and very pleased to be presenting on behalf of our group today. As James had mentioned, Dr. Kuo is in transit still due to a flight cancellation late yesterday. So I'll be talking today about a recent project we have coming out of our group looking at the influence of spousal genotype on alcohol problems in marriage. And again, this is collaborative work. I'm just representing our team here today. So the fundamental question that's underlying this project is the question of how do marriage and genetic factors influence alcohol use disorder? So we know that marriage is an important context for understanding alcohol use and the development of alcohol use disorder. And we also know from genetic epidemiological studies that there's a substantial degree of genetic influence on alcohol problems with a heritability of roughly 50%. Now, when we think about the mechanisms or the ways through which genotypes influence alcohol problems phenotypes, the conventional understanding is, of course, one of direct genetic effects. So our own genetic predispositions are influencing our likelihood of experiencing alcohol problems. But we also know from an emerging body of work that direct genetic effects represent only one potential way that genes can confer risk for alcohol use disorder and a variety of other behavioral phenotypes. And this is because in close relationships, a social partner's genotype is also an environment for the target individual, representing a social or indirect genetic effect. So the next set of animations here is just walking us through what we're meaning when we're talking about social or indirect genetic effects. So the basic idea here is that a social partner's genotype is accounting for variation in one's own outcome or phenotype. So in the context of a marital dyad where we have two spouses, each of them have their own independent phenotypes, or I guess they are typically related, so they're not independent. But when we, sort of partition the variation in, each spouse's phenotype, we're typically doing that in terms of the genetic and environmental components that are contributing to variation in each spouse's genotype. But if you look at spouse number 2 in this diagram here, we also see that the phenotype of spouse 1 represents an environment for the second spouse in our dyad. And if we follow our path tracing rules from structural equation modeling, we can trace back through that environment provided by our spouse back to the genotype of the first partner. And that's really that pathway that we're tracing in our models today. So there are a number of studies that have documented thus far sort of hints with respect to social genetic effects in spousal relationships for alcohol outcomes. So from the UK Biobank sample, individuals whose spouses carried the protective A allele of the ADH1B SNP ending in 984, they drank less compared to those whose spouses were not carriers. And in a more recent example from work that I've done with my collaborators with the Swedish National Registries, we found that marriage to a spouse with a high family genetic risk score was associated with an increased likelihood of developing drug use disorder during marriage. And this was, again, using a more latent genetic approach rather than a molecular approach. And then other work from the Health and Retirement Study recently demonstrated that a spouse's genome-wide polygenic scores were associated with BMI, smoking, and drinking behavior. So again, sort of this emerging body of evidence that there are spousal social genetic effects for substance use phenotypes. Now, some of the limitations from these prior studies are first and foremost that they've relied or sort of historically focused on alcohol consumption rather than problems. And sort of from a clinical perspective, we do care about problems, not just sort of how much one is drinking, but the degree to which it's interfering with one's life. And they have also typically relied on largely molecular genetic information using genome-wide polygenic scores as the indicator or the sole indicator of genetic risk. But there is recent evidence coming out of the Danish registry suggesting that both family history and genome-wide polygenic scores represent independent and complementary indicators of genetic liability for substance use disorders as well as other psychiatric phenotypes. So really we need to include both our latent and our measured genetic indicators. So the framing research question for our project today was whether we would find evidence for spousal social genetic effects such that marriage to a spouse with a higher polygenic genetic predisposition, either indexed using the molecular approach or a latent family history approach, would be associated with greater alcohol use disorder clinical criterion counts during marriage. To answer this question, we use data from the collaborative study on the genetics of alcoholism sample. And we defined within that sample 660 opposite-sex marital dyads. And these were individuals who were most genetically similar to individuals from European reference populations. And these were also individuals who were in mutually defined first marriages. And we defined them based on their age or the year in which both the partners in the sample were reporting on when they got married. And this sample, on average, got married on a relatively younger age end compared to sort of population-based data in the US today. And just to give you sort of the big picture, the age at last assessment for participants in the sample was roughly 50 years for men and 52 years for women. And the sample has roughly a year or two of post-high school education. So our outcome measure here was DSM-5 alcohol use disorder criterion counts during marriage, which we were able to ascertain based on the information participants provided regarding the recency and onset of their symptoms. And then our focal predictor was a problematic alcohol use polygenic score, and this was calculated using PRS, CS, and the discovery GWAS weights came from the MVP program, the MVP problematic alcohol use meta-analysis, with the COGA sample, of course, removed to remove that redundancy. Our latent genetic indicator was parental history of alcohol use disorder, and then our covariates included age at last assessment, sex, as well as the first 10 principal components. So the next figure is just walking us through some of the descriptive data and sort of, as we heard about earlier today, the spousal resemblance. And so the males in the sample during marriage had on average met under 4 clinical criterion counts, and the females met just over 1. And these are relatively high numbers to be getting in the sample, which also represents the sort of clinical nature of the COGA sample. It's a very severely affected sample. So the phenotypic correlation, polygenic score correlation, and then the parental history of alcohol use disorder correlation, these are the spousal resemblance indicators, are what's plotted here in each of those bars, ranging from 0.09 Pearson correlation for the polygenic scores, up to 0.16 for the phenotypic alcohol use disorder criterion count correlation. So we modeled our data using a dyadic perspective and specifically used an actor-partner interdependence model, which, for those who may not be familiar, treats both partners in a pair as both what we call actors as well as partners. And this is basically an extension of a multi-level modeling approach, and it accounts for both the correlation between partners' polygenic scores, because we know that those are correlated, and it also accounts for the correlation between the residuals of the spouse's alcohol use disorder clinical criterion counts. And so what we're able to parse the variation into are both actor effects, so the association between polygenic scores and one's own criterion counts. And then these cross-path effects here are the partner effects that were sort of of primary interest to our research question today. So to just simplify the diagram and the results that I'll be presenting next, I've just taken that second spouse away, but rest assured that that second spouse and the interdependence between spouses is statistically accounted for. So again, we're interested in our actor effects and our partner effects. And what we find is that, consistent with other work, that there is a direct genetic effect, that actor effect, an association of 0.23 between one's own polygenic score and then their own alcohol use disorder criterion count during marriage. And then we also find evidence for a social genetic effect. That's about 1/3 the size and magnitude of the direct genetic effect. So the next step of our analysis was to model the polygenic score and then that latent family history measure jointly. Now, the sample size for this analysis was a bit attenuated compared to our polygenic analyses, and this just has to do with some sort of historical sort of characteristics of the COGA sample and who was administered what protocols at different points in the study's history. But what we find is that, or we continue to find an effect, a direct genetic effect for one's own polygenic score, and we also find a direct genetic effect using the latent parental history of alcohol use disorder measure. And although in this model, where they're again modeled jointly, the social genetic effect or the partner genetic effect is no longer statistically significant, meaning differing from 0. The magnitude remains of the same effect as what we saw in the full sample. So our sort of interpretation of that is this likely has to do with a sample size issue and sort of imprecision in the estimate as opposed to sort of the effect completely going away. We also conducted a number of robustness tests to sort of probe the sort of sensitivity and robustness of our social genetic effect to a variety of other confounders. So in this slide here, what we're showing is the effect, whether the effect remains after we account for both the actors and the partners' educational attainment, because we know socioeconomic assortment is sort of widespread. And we continue to see an effect for the partner's social genetic effect, even after we control for both actors and partners' educational attainment. And we also, just sort of perhaps interest to the group, we also found an effect for partner educational attainment, such that marriage to a partner with higher educational attainment was associated with lower alcohol use disorder criterion counts during marriage. And so we also were interested in looking at whether these social genetic effects were stronger, perhaps among those who were more at risk. So we tested this with an interaction effect between sort of actors and partners' polygenic scores, and we really didn't find evidence for any sort of interaction. So it didn't seem like anyone was particularly sensitive to the polygenic score of their partner. So with all of those data, we also want to sort of add the limitations with which they should be interpreted. And of course, as we saw in those descriptive data, COGA is a very high-risk sample, densely affected by alcohol use disorder and other related psychiatric conditions. We had to limit the analyses to legally married opposite-sex couples, and that's based, again, on some of the historical characteristics of how marital status and marital history information was assessed in the sample. We focused here only on like-type polygenic scores, so problematic alcohol use polygenic scores, and cross-disorder spousal social genetic effects are largely uncharacterized. So this is an area ripe for opportunity. And so in conclusion, to wrap up, the study here, we hope, highlights the importance of social genetic effects for alcohol use disorder, and that there are really multiple pathways from genotype to alcohol use disorder phenotype. And the sort of need to consider beyond direct genetic effects to also consider the genetic makeup of the environment. And indeed, close relationships are these important contexts for the development of alcohol and other substance use disorders, and our partner's genes become part of our environment and part of that math in terms of thinking of the eventual phenotype. And lastly, and I think that this was sort of hinted at some of the earlier presentations today, is that there is also future opportunity for characterizing what are the loci that contribute to indirect genetic effects for alcohol and other substance use disorders. Amelie Baud, of course, has done a number of studies in this area, characterizing this within a rodent model. Obviously, we're not there yet in terms of human GWAS for direct genetic effects to be able to start doing this, but at least that's sort of providing a path forward. So with that, I thank you for your attention, and we'd be happy to answer any questions.
Do we have time for at least one question?
Thanks for the talk. I'm wondering, since I think it's intuitive to assume that alcoholism is a factor that may lead to divorce. So by constraining the analysis on married couples, I wonder if you can comment on this potential missing and not at random problem. And another question is, do you worry about the fact that the GWAS for alcohol consumption already has both direct and partner effect baked into the SNP effects, and when you construct polygenic scores based on this combined estimates, would that change your interpretation of the different edges you obtain in your analysis?
Yeah, absolutely. And so if I keep in mind your first question about, was it about the sort of divorce? Yeah. So divorce is also genetically correlated with the genes that contribute to alcohol use disorder. And yes, so certainly there's sort of a potential for bias to be introduced based on that. For this particular study, people could eventually divorce, but we limited our dependent variable to be criterion counts that happen during marriage. And actually, it turns out that your risk for AUD increases quite significantly after divorce as well, and that can depend on the alcohol use disorder characteristics of your partner. So women who divorce an alcohol-dependent men tend to have a reduction in their risk for alcohol use disorder, but for males, the loss of the relationship often seems to sort of potentiate further problems. I don't know if that answered your question, though. Yeah? Okay, and then the second part was about sort of, yeah, I mean, I think that we've seen for a number of different phenotypes, and it hasn't been done for alcohol use disorder yet, but that yes, I mean, a big part of what's being picked up in GWAS is the environment too, and that includes both features of social partners in one's environment that could be influencing behavior. So I don't have a solution to that though. Perhaps someone else in the room does.
I just had a quick question, and this is my own, I'm not sure. With the million veterans, were they trained this score? Was it mostly men? And how do you think that's affecting, is the prediction similar in women here? If you just look at, is it still good prediction in women? Just because I'm thinking of a lot of different social factors, different reasons women drink, different hormonal interactions with alcohol, all these kinds of things that, whether you think that that might have any effect on your results.
Yes, and so that the MVP GWAS was a meta-analysis across a number of studies, but as you were sort of tapping into the, so it wasn't just limited to veterans, which have their own sort of characteristics, sort of social characteristics that are also different from general population samples. But yes, I mean, if I recall from like other projects that we've done, there tends to be a stronger polygenic association in males, which probably reflects sort of like who's going into the GWAS, the discovery sample. But that's not, there's still association in women. And I'm guessing that the error bars are enough that they probably don't significantly differ, but the magnitude of the association is, I remember, stronger for males.
Great, thanks.
Thanks.
Okay, our next speaker is Sylvia Barcelos from USC. Education reduces dementia prevalence and mitigates the genetic risk of dementia.
Okay, so this is joint work with Patrick and Leandro. We're here. I'm Sylvia, currently at USC, moving to UW-Madison this summer, so very excited. Then also we have Sneha from USC, is a grad student, and our dementia expert, Ken Langa from University of Michigan.
That's the mic.
Oh, thank you. Okay, great. So the number of Alzheimer's disease and related dementias, or ADRD cases, is growing worldwide. It's a very costly disease, both emotionally and monetarily. In the US alone, it's estimated to be, the cost is estimated to be about $305 billion. With population aging and the increasing life expectancy, these trends are expected to persist, and costs are gonna triple in the next few decades. So in this context, it's really important to understand factors that can decrease ADRD risk and delay its onset. And education has been pointed as one potential factor that could decrease risk. So several studies have shown that education is associated with lower ADRD risk, but of course we all know that correlation is not causation, and there are third factors that might confound this relationship, such as socioeconomic background and genetics. The other challenge here is that dementia is difficult to measure, it's difficult to diagnose. There's not a one-to-one correspondence between cognition and dementia, and especially when we use survey data to study this question, there is pretty important selective sample attrition, right? As people become cognitively impaired, they tend to drop out of surveys. So there is some evidence, including by Jason and co-authors, that use quasi-experimental experiments to study the causal effects of education on cognition and cognitive decline. So there's some evidence that, yes, education improves cognition in later life. There's only one study that we know of that looks at the causal relationship between education and dementia diagnosis itself, and that's a study in Sweden that has some differences, methodological and context differences to ours, but they don't find an effect of education on dementia. So what do we do? we are going to look at this causal relationship by exploring a natural experiment that generated exogenous variation in education. And we're going to use dementia outcomes from administrative health records. So the natural experiment is a compulsory schooling reform, age reform, that Patrick, Leandro, and I have used in prior work, so apologies if you've seen this too many times. We use UK Biobank data. The sample size is about 90,000 people who were born around the time when the reform was implemented, or the threshold that the reform affected, right, the birth threshold. And then in addition to like kind of looking at what's the effect of education on dementia risk, we're also going to use the genetic data we have from the UK Biobank to ask the question, does education mitigate the genetic risk for ADRD? So in other words, does it reduce the association between genetics and ADRD risk? So what's a natural experiment? It's in 1972, England, Scotland, and Wales raised the minimum school leaving age from age 15 to 16. The interesting thing about this reform is that it only affected students born after September 1st, 1957. So that generated a really nice discontinuity between date of birth and education. So here I'm going to show you graphically. We have the fraction staying in school until age 16 by date of birth. This is the threshold of the reform. So again, people who were born before this date could drop out at age 15. People who were born after that age had to stay until age 16. And what we see in the data is that we see this nice discontinuity where people who were born just days before the threshold, days after the threshold, were 14 percentage points more likely to stay in school until age 16. So our empirical strategy here is going to look at the administrative data, many, many years when these kids are not longer 16, they're like 65. And we're going to ask, do we see a similar pattern in the data? Do we see a discontinuity in ADRD risk by this birth cohort? And if we do, then we're going to say that's because of extra education, okay? So the assumption behind this method is that because this threshold for the policy is somewhat arbitrary, the people born to the right and to the left of this cohort, of this threshold, are similar in other factors that would lead to ADRD risk. So one of these factors is whether you have parents who were diagnosed with ADRD. So here we're just plotting that across this threshold, and you can see there's no discontinuity there. Right? So again, this is supportive that our identifying assumption is working. Same thing with the genetics, right? So this is the genetic risk for ADRD, not a discontinuity there. Okay, so we have much more of this balance test in the paper, but I'm not going to go into that. So what's our main outcome? So how do we measure dementia, right? So we use, again, the UK Biobank. And the nice thing about the UK Biobank is that it's linked to all these registries, in particular hospital records and death registries, and also some self-reported medical history that allows us to know if, from any of these sources, if the person was diagnosed with dementia and when they were first diagnosed, okay? So our main outcome here is that AG is going to be ADRD diagnosis as of December 2022. So these, at the threshold at this time, are going to be 65 at this time. So still somewhat young for ADRD, but we're going to see, prevalence is still low, but we're going to see pretty clear results nevertheless. Okay, so that's kind of for me, that's the beauty of the administrative data, right? So even though some of these people were just surveyed by the UK Biobank in the early 2000s, we can still keep following them with these linkages. And even if they got dementia or died, we can still study them. So that's the beauty for this question we're interested in. So here's just kind of some nice checks of this administrative measure of dementia. Here is the UK Biobank also has some data on plasma proteins that have been implicated in ADRD. And when we graph those, we can see that those people that were diagnosed with ADRD based on the administrative data do have higher levels of this protein. So this is just a nice check with some biomarkers. Okay, so what do we find? So this is, again, the same kind of graph that I showed you for education, that, but now it's a fraction diagnosed with AGRD based on our measure. And you can see, again, prevalence is still low, but there is a very clear discontinuity at the threshold of the reform. So again, the AGRD risk for those born before, about 0.46%, and it drops by to 0.26%. So pretty, again, prevalence is low, but the relative effect is very large and clear in the data. So I'm going to, it's all highly significant. I'm going to kind of just skip through just because of time. It's all highly significant, but it is significantly different than zero, but the standard errors are large. So we believe, kind of like the way I think about these results is that, yes, there is some pretty clear evidence that education has a causal effect in protecting against AGRD, but I think we still need to wait for this cohort to become older so that we have more tighter estimates of what the magnitude of this effect is. So here's just a little nice graph of how, because of the, okay, my thing might not work. That's sad. Will it not work? Yeah. Well, yeah, it does work. Yeah, this is a little movie of the administrative data. So I'm just showing you this RD graph as people are aging, right? So you can see how, as they age, the prevalence increases, and this gap between the people born before the reform and after the reform starts to open up. And so, again, as we move forward and we keep following these people, we think this is going to be, a clear result. This is just like another way to show the similar result is that here's hazard rates for those born before the threshold and after the threshold, and you can see that it increases, it kind of diverges over time, and it's going to become more and more precise. So so but we're here to talk about genetics, right? So ADRD has a big genetic factor, and there's this perception: is genetics can genetics be altered by environmental factors or not? Right. So here we're going to ask: does this reform affect the relationship between genetics and ADRD? So we're going to construct a measure of genetic risk for ADRD that has both a polygenic component and ApoE genotype. And we're going to split the bottom and top half of this distribution of this measure, and we're going to see how the results vary for these two groups. So here's kind of I thought I would show this is the ADRD is our administrative data, and this is how it's predicted by the genetic. Well, this is the first one is the AD-PGI, with the compo weights without the ApoE, and these are the ApoE interactions, right? So you can see that they both predict pretty well AGRD, and they predict, and if we add both together, they both have predictive power on AGRD. Interesting, we were talking about direct genetic effects and EA and biases. Interesting, if we look at AGRD, this genetic risk measure, and we do the first column is without. So this is just, again, the UK Biobank has a subsample of siblings. So the first one is the subsample of siblings without family fixed effects. The second column is the subsample of siblings with fixed effects. And we see that there's not a lot of confounding to begin with in this AD genetic risk. So if we do the test, we can reject that these two coefficients are the same. And as Marina is going to present tomorrow, we see a similar thing when you use lifelines, like Dutch data. We also don't see a lot of confounding on this AD-PGI, which is pretty different than the EA-PGI, for example. So so I'm going to do the same thing now, just breaking by this genetic risk. So this is pre. You can see, those with low genetic risk, as you expect, have lower ADRD prevalence than those with high genetic risk before the reform. But what happened after the reform is that we see a reduction in ADRD risk for both groups, but it's much higher for the high genetic risk group, so that the gap between the red line and the black line kind of completely vanishes. So what this is saying is that Yes, so these genetic effects are more immutable, and the reform actually affected the relationship between genetics and the outcome we're interested in. Okay, so, and is this because of, the two groups were affected differently by the reform? And the answer is no. So if we look at how the two groups were affected by, the education of the two groups were affected by the reform, they pretty much overlap each other. So it can't be explained by, maybe the reform affected them differently. So just in my last minute, I'm going to just talk a little bit about some potential pathways. So I showed you pretty clear results on, education protecting, secondary education protecting in terms of ADHD risk later in life. So what happened over this 40-something years? Well, we have some data on income and health. So basically, what the reform, we see that this reform, we and other papers see that show that this reform did have an effect on income, wages, occupations. So different than the Swedish reform didn't have an effect on SES, and maybe that's why they didn't see an effect on ADHD. The UK reform did have an effect on SES over the life course of these people. The other things that we see, especially when we break by low genetic risk of ADHD and high genetic risk of ADHD, we see a reduction in obesity and a reduction in diabetes also, which again might be part of why we see effect on ADHD. Okay, so combine NACTOR experiment with dementia outcomes from other institute data. We find, again, evidence that additional schooling can lower ADHD risk, especially for those with high genetic risk of ADHD. Limitation, this cohort is relatively young. Our estimates are implausibly large and somewhat noisy, even though they are highly statistically different than zero. So it's important for us to follow up this cohort with this administrative data as they age and see what happens. That's it. Thank you.
Hello, I'm Nico from Bologna. Thanks so much for the presentation. I have a couple of questions. Maybe if there's not enough time, you can answer them to me afterwards. We can meet, but because it's a few things. The first one is like the UK Biobank. We know it's not a representative sample,
right.
So it's biased towards higher educated individuals. So I was thinking this could kind of change your sample before and after your reform you're evaluating. I'm not sure how this would bias your results. I was kind of thinking about it, but like colleagues actually of California, and of Netherlands, like Joe and Titus, they released these weights, to weight the UKB. So I haven't used them yet, but I was wondering if you have been playing around with them and if your results change. So this is one thing. The other thing, very briefly, like you're estimating, I understand, like an intention to treat. I was wondering, I'm not sure if you've done it or maybe you didn't understand it well. Could you kind of do a two-stages least squares, kind of using education and kind of getting a Latin, a bit more, maybe a precise estimate?
Yeah.
And the third thing is like because this is a personal preference. Like I'm very much interested in the mechanisms. Like if you would see like something about kind of labor histories, if like we do have a lot of information about labor histories in the UKB. If individuals actually move to some occupations with, for instance, more kind of cognitive.
That's interesting.
Like this was part of my thesis, therefore I'm kind of interested. But yeah.
Yeah. So yeah, all good questions. I'm trying to like go quickly because I know there's a few other questions, but. No external validity, but we show in the paper a lot of internal validity. So, right, like it's not, the internal validity is right, maybe the application to the general population, not. But we still, I think, because we're not really putting a lot of emphasis on the magnitude anyway, it's more like the result. But yeah, we could use Titus's and Schwartz, things to recalculate. This is the two-stage squares. I didn't have enough time. And we looked at, like, in order to get, like, more precise, we looked at people who dropped out at age 18 or younger because the reform didn't really make them stay up to 18. And it's a little bit less, more precise there, because we're focusing on the, but it's still implausibly large, right? So the mean is 0.6, so it's still very large. And the last question, yes, Leandro has a paper on occupational history, but we definitely see that the people moving to a better occupation. So it's definitely, it's a kind of like, it's a huge effect, but it's also, you need to think that this is a lot, it's something that it's, yeah, so it's kind of interesting, yes. Yeah, so maybe we should add more into that in here, but yeah, that, you're right.
Thanks so much.
Yeah.
Let's take one more question.
Hi. Oh, is this on? Okay. So I had a question. I know that we're looking at staying in school just 1 year longer. Would you expect that with continued education, even into college and past 18, we would see potentially even more of a decrease in Alzheimer's risk? Okay, or I was just wondering if this could potentially be like a sensitive period that we really need them to stay in school for at this time, or if we would really
I don't think we know.
Yeah.
Because I really like what we're doing here is like, how do you say, looking under the light, right? The experiment gave us a light and we're looking like doing the best. So this is kind of why we're looking 15 to 16, because we have this experiment, right? I don't think we know enough about other levels of education. The reform itself did not affect over 18 or college going. It did affect, in the UK, they have this educational qualifications that people take at age 16. So that are like O-levels, A-levels, the reform did affect those, and those are valued in the labor market. So this is kind of another thing that sets us apart from the Swedish reform that didn't have an effect on, that didn't see an effect on ADRD, right? So there's definitely an effect on labor market outcomes from this reform that kind of evolves as people age. Yeah. Thank you. And I'm happy to answer more on the bridge. Thank you.
Okay, and then our last presenter on this panel is Giorgia Mazzetti from the University of Bologna. She'll be presenting on the role of gene-environment interaction in the gender wage gap.
Hello everyone. First of all, I would like to thank you, University of Wisconsin, for hosting me and also doing this wonderful presentations conference. It's really interesting. So I'm a second-year PhD candidate at University of Bologna, and yes, I'm presenting my work on the role of gene-environment interaction in the gender wage gap. So my motivation and the subsequent research question is that gender wage gap is a pressing social issue, but it has never been studied through the point of view of gene-environment interaction, although we all know that economic success has a genetic basis. And so genetics is a gender-neutral measure since it is made with the first 22 chromosomes. And so my question is, do gender norms suppress genetic potential? So specifically in this paper, I will look on how the labor market rewards genetics differently in between males and females measuring hourly wage. So I will use the health and retirement study. So it is a panel starting from 1992, an unbalanced panel going still ongoing, and I will focus. So they made interviews each 2 years, and I will focus on people currently working at the time of the interview, and among them, on the one that are part-time or on the labor force status, and of course with genetic data. So I'm talking about around 12,100 people. My contribution would be on the literature gender wage gap, since it is has not been studied through the point of view of genetics, and so this will be studying gender discrimination in terms of lost genetic potential, as there are papers on stereotypes suppressing potential, for example, in math gap or STEM gap and so on, but also in genetic literature, since gender wage gap has not been studied, and a similar study has been done by Pamela Herd on educational attainment in 2019, and she found a varying association between EAPGI and years of education. So this is just to give an insight of how data looks like. So this is a polynomial approximation of hourly wage on the EAPGI. And we see that there is a much steeper line for male compared to female. So this is a first hint that we might have varying associations, so something might go on in the environment. So first of all, I look at labor force status. So these are probability estimate, linear probability models. And so one standard deviation increase in the EAPGI increases the probability of being current working at the time of interview of around 3%, and 2% of being employed full-time. But we see that when interacted with gender, the effect is almost halved, or even more. So the decrease of around 1.5 and 1.7%. So this is my baseline empirical specification. So omega I would be log hourly wage. I use logs so we can interpret coefficients as a variation in percentage. I regress it on gender, genetics, of course, the 10 principal component and the interaction. And the covariants I'm using in the baseline are age, age squared, and mother's education and father's years of education. And what I'm interested in is the coefficient beta 3, since its significance and sign can give insight on varying association between across genders. So these are my estimates. In the blue one is the baseline model, and we see that there is a one standard deviation increase in EAPGI increases your hourly wage of more than 10%, around 12%. However, when interacted with gender, this decreased by 3%. So around 1/4 of the magnitude is cancelled out when you are a female. In the second specification, I add the year of birth and the region of birth, and the estimates are pretty similar. But what I find remarkable is that in the third specification, I add years of education. So we see that the effect of EAPGI is pretty much reduced, as we could expect, but the interaction increases and also is more significant at 5% compared to the other that are significant at 10%. Possible mechanisms or pathways, I look at job occupation. So on the left, we have the 3 most paid jobs. So we see that we have a negative interaction in female, as we could expect in the literature due to glass ceilings effect. A one standard deviation increasing EAPGI increases the probability of working in this high-paid job of around 5-7%. The interaction is negative, but as you can see, it's not significant. I think this might play a role, but the sample size does not give me enough power. On the other side, on the right side, there are the least paid jobs. And what I see is that while the EAPGI seems to have no effect on being employed in these jobs, the interaction with female is negative. So this rather exacerbates the gap rather than explaining it. And we see that the probability when you are a female, one standard deviation increase of EAPGI decreases the probability of being employed in a low-paid job of around 2%. I also look at potential mediators. So I looked at personality traits, but the only one I find significant is conscientiousness. I also look at self-employment. So this is a dummy. We see that female are less likely to be self-employed, and the interaction with EAPGI is negative, while the relationship between being self-employed and wage is positive. Years of education on the other side might be a potential mediator. This is a whole old cohort, so we know now that the effect is reversed, as also Pamela had showed, but I mean, these people were born a lot of years ago, so it is, we could expect something like this. So in my future steps, I would like to merge these with administrative data to see career path, and I will also like to use ad health dataset to see the evolution of the gap over time. And of course, I would like to exploit an exogenous variation to try to infer more, give insights also for public policies. So in conclusion, I think genetics is a really good measure a really good counterfactual when we measure change in environment between males and females. And we see from here that the relationship between the EAPGI and the wage is much weaker for women compared to men. And so this could be an insight for gender discrimination. And yes, I look personality traits as well as job occupation might play a role. Thank you very much for your attention.
Thanks, Georgia. Steve Blair, Queen's University. I'm going to wear my labor economics. Sorry, Steve Blair, Queen's University. I'm going to wear my labor economics hat, so everyone else forgive me. So usually when I'm thinking about estimating a gender wage gap, the most important thing is defining equal work, right? So like that last point that you had, job occupation, I think is really is key. But I think as you're going forward, the data sets you're using are sort of going to be hard because they're both going to be working in worlds where there's a lot of decentralized bargaining on wages. So I'd strongly encourage you, if you can, find a data set where collective bargaining plays a bigger role. Because that is where you can actually go within job, within bargaining agreement, and then you can see if there's gaps, and then you can see what's the remaining role, because otherwise.
Yeah, it's totally true. Up to now, in the public HRS data set, I just have like job occupation according to a census. So, but I'm planning to apply for restricted use data of HRS to delve a bit more deeper in this matter. Yes. So thank you very much.
I think you can speak for, I don't know. Yeah. So another matter that's really important for the wage gap we know is children, right? So I guess everyone in your sample, especially they are older, would have had children. Do you know when you measure these wages? Is it near childbirth, or because that's maybe something you can control for?
These are measured at the time of the interview for your current job. So they all have had, the majority have had children. So if I insert this as a control, it actually doesn't change the results. But of course, I know that child penalty is a really great matter, and that's why I would like to use career path. So with the year of birth of the child combining with all the employment history data, I can see also how this infer. But at the time of interview, yes, it is asked when they are at least 50 years old or something.
So the children already grown
up.
Yeah. The other thing I was thinking of, did you look at so you're looking at the incentive margin in terms of the wage. Did you look at whether or not they are working, the 0-1 variable?
Yes, I look at. No, there are a lot of women that are not working. I look into the labor force status, and I see that yes, the likelihood of being of working is higher for high PGI. But on the considering the possible genetic selection, I do not see in the sample of having a wage, of when they report a wage. So people currently working with a dummy, yes or no, is a much higher sample. But for this analysis, they have the same PGI distribution. But yes, in general, a higher PGI increases your probability to work, and this is less strong for women. Yes.
Really nice presentation, very interesting. I actually kind of wanted to maybe slightly push back on something you said with respect to genetics being gender neutral. I guess heuristically, I can see why you would say that, why we would believe that. But I think the way that we're working with genetic data is probably not gender neutral because, for instance, you're looking at the PGS for EA. Is it possible that there could have been gender bias baked into the GWAS they performed on educational attainment, where it could have been sort of expressed at the very level of gene discovery? So I suppose maybe if you could comment on the possibility that even the genetic index that you're using could have already been influenced by some form of gender bias.
Yeah, so I state this because I looked at the GWAS, the latest GWAS made by ISU, and she says that the contribution of the X chromosome is negligible. So up to now, the index predicts, it should be gender neutral since it does not interact with the X chromosome. Of course, so that is why I attribute the different predictive power due to environment rather than biological mechanisms.
All right, thank you so much to the panelists. Let's give them a round of applause. And I think we'll move on to the next one.
So we'll move right into the next session to stay on time. So this frontier session is both a potpourri session where where you get submissions and some go together really well, and then you have these frontier elements of both, like how do we get the data that we get? So what about the willingness for participants to actually contribute to their DNA? What are researchers doing with the data, which are the second one on social networks of social genomics researchers, and then finally gene expression, which I think is still upcoming. We see 1 or 2 papers a year, but it's still, I think, an emerging area that's of very high interest. So with that said, we'll start these 3 papers, and Manisha will start.
Hi everyone, my name is Manisha. I'm a graduate student here at UW, and I had the fortune of working with my co-author Richard. Some of you might know him on this project. Sorry, I have my laptop over here. It's just because I'm blind and I can't see anything on the screen over there. So this project is about the willingness to donate DNA for science. Maybe some of you have donated DNA for some sort of a research study, or these consumer testing services like Ancestry.com, 23andMe. But this is related to the Dutch population. So So as we all know, genetic data, genome-wide data, is becoming extremely widespread for use in research, healthcare, and consumer testing, etc. There's a massive worldwide effort to collect genetic data. Governments are putting in millions of dollars for this. And as we all know, there are valuable applications of this in social sciences for establishing causality, to use as instrumental variables, proxies, etc. So one thing about these studies is to see whether the individuals who are donating data are representative of the entire population or not. For example, in the UK Biobank, 9 million invitations were given out, and the acceptance rate was less than 6%. The volunteers were healthy, educated, female, and non-immigrant, so not representative of the entire population. And this sampling bias creates statistical issues. So it creates the problem of biased estimates, or a loss of power, and this is extra problematic for gene environment or GIV studies. The motivation of this paper is therefore to study this decision process of individuals about donating DNA. And this is desirable because of 3 reasons. It can help improve the recruitment process or prevent selective attrition. It can reveal sources of bias that can be adjusted later in empirical research. And it can be used to identify variables for stratified sampling, for example, using weighing. So in the present study, we look at willingness to donate DNA for science. focusing on three things: incentives, preferences, economic preferences, and socioeconomic characteristics. So just to make it clear, like the sample of DNA was not actually like collected in the study. This is a prospective collection. So the individuals were asked, would you be willing to donate DNA? After presenting like some information, but no actual DNA sample was collected. They were said that, like they were told that the donation would be using a saliva sample for non-commercial use. So as I said, this is looking at a representative panel of Dutch households. The survey was conducted in November 2020, and it's perhaps the first study to study the characteristics of non-responders as well. The invitation was sent out, the study invitation was sent out to about like 6,800 individuals, and the response rate was about 78.5%. So the individuals who were presented with this survey, they were shown like two experimental treatments. So the first thing they were showed was some information on the risks and benefits of donating DNA. And some of the individuals were randomized to get like some sort of an extra information, just some more information on the risks and benefits. And some of them were presented with just some baseline information. And the second randomized treatment was the amount of hypothetical monetary award. So how much money you would get if you hypothetically donate DNA. And again, like individuals were randomized into these three groups. So the first was 10 euros, then the second 20, and the third 50 euros. In previous research, for example, like in the Your DNA, Your Say project, they looked at the willingness to donate DNA and medical data for different users, such as medical doctors, for-profit researchers, and non-profit researchers. In 36,000 people in 22 countries, they found that the willingness to donate was 50% on average, but 67% in English-speaking countries. They saw that the willingness was about the same when compared to like donating for medical doctors or non-profit researchers, but lower for for-profit researchers. And greater willingness to donate was associated with being younger, more educated, having children, having a Western background, and having familiarity with genetics. So in the present study, in this project, the individuals were asked how likely is it that you're going to donate DNA based on the information that's presented to you. And the information, as I said, was two things. First, the information about the risks and the benefits, and the second was the amount of financial incentives that they could have gotten. We dichotomized, so they were presented with a scale of 1 to 5, 1 meaning they would certainly donate, certainly not participate, not donate DNA, and 5 meaning they would certainly donate. And we reverse coded this and dichotomized it to get a binary outcome. According to this binary outcome, 53% of the individuals were willing to donate. We also looked at the German SOEP-G panel, where they like had actual donations of DNA, and the willingness to, and the donation rate over there was 57%, which is quite comparable to this study. So we look at stated preference measures of economic preferences. So patience, risk, positive and negative reciprocity, altruism and trust, which correspond to the global preference survey. And we also looked at some revealed economic preference measures. But in the end, like we just decided to focus on stated measures because the revealed measures did not correspond very well with the stated measures, and there was a lot of missingness over there. We're using a logistic regression. So we didn't add all the variables at once in our regression equation because there was a lot of like non-missing, there was not a lot of overlap in the non-missingness in variables. So we have a baseline model in which we have just demographic characteristics and the experimental conditions. And then we add like these different domains such as stated preferences, revealed preferences, personality, health and lifestyle, and attitudes and opinions about DNA donation and government to like these demographic variables one by one. We're using inverse probability weights to adjust for non-response and heteroscedasticity, consistent, robust errors. So for the main models, this is just like selected models and selected variables within each model. I'm just like highlighting the variables which were like significant and also the incentivized like conditions. So what we found was that if you look at model 2A, we found that the stated risk willingness was positively associated with a greater willingness to donate. That's this. And then we also found that the extra information condition was not statistically significant, but the incentive condition, getting more money, 50 euros as compared to the baseline of 10 euros, was significant, but when we look at the average marginal effect, it was not that much. Other things that were like important was extraversion. So this was also positively associated with a greater willingness to donate. Self-rated health, so individuals who are healthier have a higher willingness to donate. And then importantly, it's whether they believe that the government can store DNA or not. So as I said, the extra information condition was not statistically significant. The 50 euro incentive was strongly significant, but then when we look at the average marginal treatment, average marginal effect, it had a modest effect. And we did a cost comparison. So suppose like all the individuals in our sampling frame, which is 6,800 individuals, get the incentive of 10 euros. We computed the baseline probability that they would be willing to donate DNA. And then we figured out like what's the cost that, what's the like number of people who would be willing to donate, what would be the cost associated with this. And we did this for the other like monetary incentive, the 20 and the 50 euros too, just to get some sort of what is the cost per marginal participant for giving out like these extra benefits. And we found that these they are diminishing returns to giving more money, and therefore it's not very cost effective. So the two main variables that we found were first, stated risk willingness in terms of the average marginal effects. And the second was whether the individuals believe that the government may store DNA. So in this figure, what I'm doing is I'm plotting the predicted probabilities for these different groups. On the x-axis, like they are these individuals based on their stated risk willingness, 1 being very risk averse, 10 being very risk loving. And within each category of stated risk willingness, there are these like 3 categories depending on whether the individuals believe the government can store DNA or not. And we predict that for individuals who are very risk-loving and who believe that the government can store DNA, almost about 75% of them are willing to donate DNA. And then there was also this open-ended question in the survey. So for individuals who were certainly not willing to donate DNA, they were asked to respond in a text whether like what's the reason that's holding them back, and we did a text analysis of their responses using frequency counts, and we found that the major concern over there was concerns about their privacy and risks. To conclude, we found some concordant evidence with previous studies. We found that the willingness to donate was low, and all the previous studies also have willingness to donate in the range of 50 to 60%. We found that the association with stated risk, willingness, and the confidence in government and societal institutions was high. But we also found conflicting evidence with previous studies. So in our study in the list, and we also replicated our results in the SOEPG panel, we found no association with age and negative associations with being more religious or having higher education. We found that financial incentives had modest and diminishing effects. Risk willingness is very important, and therefore lottery-based rewards are inadvisable. And researchers should just consider these participation biases in genetic research data moving forward. Our recommendations are that more funds should be allocated to data security because privacy seems to be an important concern for these individuals, and also to improve communication materials when displaying information about the risks and benefits of the study. Yeah, that's all. Thank you.
Thanks for the presentation. I just wanted to mention that the Understanding America study, that's an online panel at USC, we did something very similar. So, and I think their data should be publicly available. So you may want to sort of do it with their data as well, and see what is consistent and what's not. I don't think they're using for any papers. So.
Yeah, I will look into that. That's a really good suggestion. Thank you.
A couple of questions, but this is more from an econ angle. So three things I found surprising, and so do you want me to start more on the positive side or the more on the negative side?
Sorry?
Okay, so I was surprised you didn't show us a table of balance at all. So at the end of the day, this immediately jumped up when we're looking at the results. Is it really a lack of balance that's driving it or not? Especially when you're doing this inverse probability weighting, right? So usually you're going to be doing, so if hopefully the groups are, if randomization worked, then there should be a really big point for doing that inverse probability weighting.
I see.
And so this kind of, that was the red flag, that was the most negative. The other thing which jumped at me, and this is just probably a disciplinary thing, is but unless you're assuming like homogeneous treatment effects or constant treatment effects, why are you doing a nonlinear estimator? You can linearize it, and that way you're allowing for heterogeneous effects, and that's probably what would be more interesting.
We could look into that, yes.
Yeah. It'd be easier too.
Yeah.
So.
Yeah.
So I was just thinking about, I feel like there are different studies and there's a lot of variability in terms of how, so like UK Biobank, like you said, only like 5% participated, but I think like for the HRS, it was pretty high. I don't remember what the numbers were, but it was like 90% who are willing to donate. I was wondering if you had thoughts about whether your study is more of an HRS or more of a UKB and like why that is.
I think it's more of a UKB study because of so I think HRS is focused on older individuals, right? I think this study sent out invitations for all individuals age 16 plus, and the panel was supposed to be representative of the Dutch population. I'm not very sure of what the the criteria for sending out invitations in the UK Biobank was, but I assume that it was not that restrictive on the age, at least.
It is an online panel, right?
Yeah, it's an online
So
panel.
it's the UAS of the Netherlands. So are they planning to, was this just a willingness, or are they planning to actually genotype people?
I think the plan was to genotype people at some point, but I'm not very aware of the discussions involving that. But I think the motivation of the study did come from that they are thinking of actually collecting data, these genetic data in the future, so it might be helpful to study who would be willing to donate, and then somehow correct for that when they actually start collecting data, but I'm not really sure of the timeline involved over there. Yeah.
Thank you.
Yeah.
Great, thanks so much.
Thank you.
Matthew's next.
Is this just forward and then back?
Yes. And there claims to be a laser. So here's the mic.
Okay. The image looks a little bit different than what I expected. It's more than fine. So, hi everyone, my name is Matthew Kuchmanich. I am a second-year PhD student at the University of Iowa in the Department of Geographical and Sustainability Sciences. I'm a geography PhD student, essentially what it means. I have no conflicts to declare, but I'm going to be using some unpublished data that's part of a larger study, so please don't take any pictures or share results without permission. Before I jump into things, I wanted to first give an acknowledgement. So this project is, or this research is part of a larger project, like I mentioned, the Beyond the Medical, the LC of Polygenic Scores. So I just wanted to acknowledge everyone that's on the team, and specifically the people that were involved in the horizon scanning process, which I'll describe the methods, and then Anya later will give more detail about that. So I would also like to acknowledge Youngren Shi, who's at the University of Iowa, where this project was first a final paper for his class, and so that got then turned into this, and he helped me in that process. And then we also want to thank the Mayo Biobank. So first getting into things, this is, I guess, a bit of a potpourri, a little bit different of a presentation than a lot of the other ones that we've had so far. So the question is, like, what is sociogenomics? And so there are a few different definitions by different people. I have them listed here, but largely they involve the combination of genetics and sociology in some ways. Here's some quotes, but like a common one that's in a lot of papers is the genetic revolution may well be underway, but the social genomics revolution is just getting started. Recently, a new group of sociologists, political scientists, and economists have joined forces with statistical genetics to make serious arguments about the utility of genetic information toward understanding social dynamics and about the right way to get that information. So I'm just putting that out there just to kind of emphasize the interdisciplinary aspect of this field and how it's bringing together a lot of different people. Now, when we think of science as a whole, scientists are important members of society. We produce truths about information, we disseminate knowledge, we influence policy. But science is also a network of people. We come together at these conferences, at other places, we talk about our research, and as such, it's never neutral. Jesnoff has the somewhat famous article that discusses this, and that political and social context shape research outcomes. So whether that be grants, other information, science is always influenced in some way. At the same time, there are disputes over truth. These represent contested boundaries, as Jesnoff writes. In a lot of ways, we have different ways of producing truths, and we see some of that even now, even today, where there are discussions about what is what and how are we using different methods to think about different phenomenon. And that begs the question of how is the field of sociogenomics growing? Who are the people involved in the research, and who is studied? So in this presentation, I'm going to discuss the first two questions through a network analysis. So to back up a little bit, part of the Beyond the Medical project, the first aim was a horizon scanning project, which horizon scanning in a lot of ways is similar to a literature review, but it's different in that it scans the horizon for upcoming literature in different methods and changes the inclusion and exclusion criteria based on information that's on the horizon, things that are coming in a way. And so I'm going to focus in on the first two aspects of identification and filtration. And that way, this is going to look a lot like a more traditional literature review. So here is just a diagram describing the process. So we used all these different databases and specific search terms related to sociogenomics, social genomics, and then those were filtered, I'm using different terms. So those were searched in these databases, and there were 938 articles that were pulled in. From those 938 articles, they were filtered according to social traits, and then 423 articles were identified that specifically focused on social traits. Articles that discussed exclusively behavioral traits were eliminated from that original 938 number. Then I brought in those articles into Zotero to pull out the authors using the DOIs. So the DOI is just a specific address that's given to most articles that you can pull author information from if you bring it into something like Zotero. I then did a complex process of cleaning and anonymizing the data, where I assigned pseudonyms also to all the authors. That was an interesting process, just because different journals have different, slightly different names for different authors. But what that resulted in was 386 total articles. So for the networks, you have to have at least 2 people that are on the paper. So any single author papers were eliminated because you can't have a network formed by one person. And in total, then there were 2,736 unique authors that were formed from that process. And then I used Gephi to create the networks, or to visualize the networks. So to get into the network connections, so these are some diagrams that show the basic idea. So author A and then author B have one connection between the 2 of them here. They might have written more than one paper, but for the analysis you'll see in a bit, this represents at least one connection. Now the same thing happens with 2+ connections, where author A and B have written 2 or more papers together. And that continues, this process, up until 10+ connections, and that's the final limit that I looked at. So then, at the same time, I looked at the top 10 authors for each of those connection categories. And so I'm going to show you those tables, but all authors have also, like I said, have been given pseudonyms. So when we look at 1+ connection, these are those authors. So Eileen Janssen has written 807 articles with at least one person in this database, and that moves down throughout Louise Anderson, who's written 569. And that's what this looks like. I don't know how well you can see that. It's probably really difficult. But that's what that network looks like. The islands are papers that people have written together, and the people that have more author connections are going to be towards the middle, so they're gravitating towards theirs because they're distributed, I don't want to say equally, but between all those different articles that are there. So then, kind of skipping a little bit ahead, I had tables if you're interested for the other connections that I have, but if we look at 5+ connections, we can see that significantly drops off. We see that Sarah Brown has written 42 papers with at least 5 unique people. And that's what that looks like there, that network. So it's a bit different, where we have, sorry, the images are really hard to read. I can't even read the bottom there. But you can see Sarah Brown has a cluster of articles, and then the top 10 in those lists, those are the ones that have labels. So there are a few different clusters where we're seeing those authors. When we look at 10+, it decreases greatly, but you can see that Sarah Brown has written 8 papers with at least 10 or more different people. And I want to say, look, so here's that graph. I want to say that the greatest connection was with 19 connections that were there. So someone has written articles with, has written articles, I'm messing things up, with 19 times with one person. So I also then looked at the websites for these top authors. There were 32 overall from all the tables, so it's a smaller number when we think about the top authors, but I pulled then their demographics from their school or other academic websites. And so when we look at this, thinking about the definitions that I first brought up, the majority of these top authors they have their education or training in psychology. Next was epidemiology and molecular biology, and sociology was actually quite low in terms of the percentage of the overall top authors. When we look at what they're working in, behavioral genomics and genetics dominates it, and then we see that epidemiology and statistics follows next, and then molecular genetics. So that molecular genetics I define as if they're working in a wet lab, and that was their primary source of research, and then developmental disparities was last. Also, in terms of their role, so primary researchers were the greatest, so I define these as people that are primary investigator, have an R01, have their own labs. Study directors were next, so these are people that have multi-site studies or studies that combine multiple departments at a university, so those are study directors. But research associates, so people that are not listed as primary investigators but are doing a lot of the statistics on research, or are don't really have clear indicators, but they're showing that their education is on analysis, or their work is on analysis. That's what the research associate is, or also if they're listed as some sort of associate role. And then last, when we look at regions, we see that North America dominates those papers. Europe comes next, and Australasia is last. I can say that North America, that of that, United States dominates that group, and then the Netherlands dominates Europe. So just as a discussion, what was interesting from this is that psychologists and not sociologists are dominating the field. This could be in part due to the history of behavioral genetics as being a bit older than sociogenomics. In the horizon scanning process, traits were, though, limited to social traits, so we eliminated articles that were explicitly on some sort of behavioral trait. With that said, a lot of studies looked at multiple traits. So if if it was on educational attainment, it might have had some other behavioral trait that was tied to it as well. Many of the top authors are study directors, so they're going to have more connections overall. And a lot of them are given authorship because their data was used in some ways. So that's definitely biasing it. It doesn't mean that sociologists are not having an influence on the field, but that these are the networks that are formed from there. Looking at top authors also allows us to think about influencers in the field. In science and academia, we use publication as a measure of success. And it can help us to understand who these leaders are. It can help guide future directions of research, thinking about how this is distributed, and also help inform the ethical, legal, and social implications of sociogenomic research. Now, there are some limitations, of course. Top 10 is an arbitrary number. I could have picked top 20, top 50. Doing that manually and looking up these authors is more time intensive. So definitely it's something it could be done. In of itself, data cleaning was extensive. So some of the authors, they could have been listed in these different ways that I have up here with spaces, or different middle names listed with periods or not. Sometimes not at all. Some people, just their last name is listed. So in part, that cleaning has a certain art to it, if you will. The categories that were there were created by me. There's no specific guidelines for those, so that's limited as well. So I guess just to conclude that this is our team, the Beyond the Medical team. This is a QR code if you're curious about other research that we do. And thank you. Hi.
Thanks for your talk. This is really interesting, and I love networks, so this is cool. So a couple of things. So I was a little bit critical of the defining social traits, like sociogenomics as the study of social traits. Like I would think of a study of, say, obesity, where you're looking at social mechanisms that affect that, like as a sociogenomic study.
Mm-hmm.
So maybe that's something to reconsider. But another, just a small, maybe analytical note, is that when you're removing isolates or when you're removing people who have, fewer than 5 publications, or for whatever the cutoff is, it might be interesting, and just for the sake of being open science, like just including a logistic regression of like who are those people, who are you dropping? And Jim Moody has, a sociologist of networks, has some interesting work on that. So it might be useful to do that just for completeness sake, so we know who we are looking at.
Mm-hmm. Okay. Yeah.
Super interesting.
Yeah.
Yeah, thank you. It's not really a thing in my field, but I'm under the impression that having too many co-authors is a thing that's frowned upon in some fields. I wonder if this could explain part of the differential finding across different fields you showed in some of your figures. And another thing is, I thought I had another point, but I've lost it. Maybe I'll come back.
Yeah, but I think the paper with the most authors had 289 authors listed on it. So, and in some ways, these are people that have come together and decided that this is our paper that we're publishing. But yeah, I think it can be limited in some ways. I was also thinking throughout the process of how, when kind of what you were saying, if I change certain thresholds, which I was thinking about, but I didn't end up changing it. You have 2 authors that are writing together, and that might be looked upon. If I were to eliminate that, that's eliminate connection and possibly kind of destabilizing the network in some ways that I've produced. So
yeah, and the second question, now I remember, is can you elaborate a little bit more on why this can be informative on ELSI issues? Because I think that's one of the bullets you.
Yeah, so I think if we're looking at how the field develops in some ways, and we're looking at these top authors, how they're influencing the field, can be certain practices or certain research areas that can be normalized that might be a bit problematic, and then through a process of normalization, then not statistical normalization, but meaning that it's formed into a norm that then it becomes more acceptable, which is debatable. Then does this research become ethical or not? But I think if you can see how the trajectory of the research, if it's focusing on one specific area, if it's focusing on one specific trait, that's a little bit more controversial or not. I think that's how it can influence the ELSI, if that's making sense.
So it seems like, related to that point, it seems like you cast a pretty wide net in terms of publications that you considered. But you could imagine that you have one person who publishes 30 papers a week and posts them to bioRxiv, and then another person who publishes 3 papers a year and they all get into science. Mm-hmm. And it seems like in terms of which person is actually having a bigger influence on norms, the science person seems like it would be bigger. I was wondering if how robust some of these clusters and kind of key figures are to looking maybe at high prestige journals or things like that.
Yeah, no, and I could definitely weight it in some ways, so that way that's part of it. But I think for this, and just as a preliminary look into it, wanting to see just how those networks were in themselves, and just the idea of connection being that role of power of who I'm writing with, rather than where I'm writing and how I'm directing my own power. So it's created through power. It will be created through networks and not through where I'm directing my energy.
one alternative, so I like this, and part of my point was already been raised about the definition of the social versus behavioral trait. There's no one way to do it, of course, but I thought of an alternative. I wondered if you would consider of insisting on the co-authorship patterns in these papers being of some set of disciplines, which is to say, can papers that only have 3 individuals who are all, say, in bioinformatics, but they're looking at educational attainment as the outcome, is that social genomics?
Yeah, I think that gets to the inclusion exclusion criteria for the studies, how we went through analyzing it in the horizon scanning process. We did have 2 people review each article to be included as well. So that was part of that 400 number that was listed there. So we did have a robust process of deciding if this should be included or not. So and just one factor of that is whether it was a behavioral trait or a social trait and how we determine that. But Anya can speak a lot more to that than I can. So yeah.
Thanks so much. That was great.
Yeah, great.
So pointer, like.
Okay.
So.
All right. Let's give it a try. Okay, it's working. Good afternoon, everyone. My name is Effie Zhiyuan Yu, and I'm currently a research associate at Johns Hopkins School of Nursing. And in about 2 months, I will be an assistant professor at University of Pennsylvania School of Nursing. But most importantly, I'm a very proud 2-time Badger, and I get my PhD and BS in Madison, Wisconsin, here at School of Nursing. So really good to be back. And so social genomics is still really new to me. I still consider myself more leaning towards the social side versus the genomic side. So being at this conference is definitely out of my comfort zone, and the word frontier is making me a little bit nervous. So hopefully I live up to the expectation. So, and also appreciate the opportunity to share what I've learned so far, and looking forward to learning more from all of you who are the best at this field. So a little background about me. My research focuses on understanding the intergenerational transmission of adversity and trying to find ways to prevent this transmission and mitigate the deleterious health impact. And understanding that adversity could be biologically embedded and transmitted to the next generation, I've obtained a K99 during my postdoc to understand more of the biological side of the story. So, and when I'm leaning more towards the biological side of the story, I'm more focusing on the immunological pathways using a social genomic framework. And the work I'm presenting today is not my K99 study, which is ongoing and involves some similar analysis. And it will also include positive childhood experiences. So just a little bit of context about this study. I actually met the PI, Nicole Letourneau, at the Child Development Conference last year, and she was presenting the positive impact of attach. And we had a conversation about whether she has involved ACEs, average childhood experiences, in their evaluation. And this conversation inspired us to do this additional analysis using their existing pilot data. So this is our disclosure, our funding, and we have no conflict of interest to declare. And so also thanks Matthew for giving a definition of social genomics. So for our study purpose, we're interested in looking at the intergenerational impact and whether the impact could be mitigated by a parenting intervention. And specifically, we want to examine whether maternal childhood average childhood experiences is associated with increased inflammatory gene expression in mother-child dyads, and whether the attached parenting program could moderate this association between maternal ACEs and mother-child inflammatory gene expression. So for background, I'll walk you through some concepts of ACEs, the conserved transcriptional responses to adversity, and brief overview of the parenting program called ATTACH, which focuses on improving parents' reflective functioning.
So when I say ACEs, this is referring to potentially traumatic events that occur before age of 18. And it could include child abuse, neglect, and household dysfunctions, such as living in stressful household environment, such as domestic violence, mental health illness, and substance use. And we know that ACEs are strongly associated in a dose-response fashion with some of the most common, costly, and serious health conditions, including 9 of the 10 leading causes of death in the US. So we also know that growing studies have suggested that immune system dysfunction, including chronic inflammation, may be one of the key biological mechanisms through which ACE affect illness or poor health in later life. And specifically, ACE could lead to prolonged activation of the biological stress response system, such as the sympathetic nervous system, increasing inflammatory burden. And that in turn may get under the skin and elevate a risk for poor health and morbidity well into adulthood. So given the mediating role of immune regulation, identifying factors that could buffer the effects of ACEs on inflammatory biology may be helpful to mitigate the risk of illness among children and family exposed to high levels of adversity. So one indicator of inflammatory activity is the conserved transcriptional response to adversity. So these are data from a molecular epidemiology study where Dr. Steve Cole and his team were looking at leukocytes or white blood cell expression activity in terms of RNA transcription in a small number of people exposed to one of the epidemiology's most toxic environmental risk factor, which is loneliness. So in this heat plot, each row represents an individual, and each column corresponds to one of the 209 genes that are differentially expressed. So the right intensity indicates relatively high rates of transcription or overexpression or upregulated expression, while the green intensity indicates relatively low rates of transcription or under or suppressed expression of genes in that person's white blood cells. So all these red and green blocks you're looking at really tells us essentially there are 70 or so genes that are systematically more active in the white blood cells of people exposed to high level of loneliness, and about 130 genes systematically less active when, and they actually represent a few functionally coherent clusters. So for example, if you look at the people who are lonely in the top section, prominent among genes that are more active, there's a block involved in inflammation or the body's first line of defense against wounding injury and the initiation of immune response. And then among the genes that are less active in the people who are lonely, the genes involved in are the genes involved in the defending cells against intracellular pathogens like viruses. So the CTRA essentially captured this transcriptional shift or redeployment of immunological resources, which is characterized by this downregulated or suppressed expression of genes in type 1 interferon response and upregulated pro-inflammatory gene expression. So since then, Steve and his team has found that this thematic shift in gene expression wasn't specific to loneliness, and they have found similar kind of profiles taking place in variety of adverse life experiences, including early life adversity. And there's a recent paper showing that there is a dose-response relationship between children's exposure to ACEs and elevation in pro-inflammatory as well as type 1 interferon expression, which is consistent with a skew towards pro-inflammatory activity. However, the CTRA, whether the CTRA manifests in younger children and what might lead to a reduction of this CTRA profile are still unknown. So the good news is that sensitive responsive parenting could be helpful to buffer the effects of ACEs on children. But the bad news is that parents' own history of ACEs can challenge their ability to practice these parenting behaviors. And we have known that studies have shown that mothers' ACEs can disrupt their emotional regulation, their parenting behavior, parent-child attachment, so essentially their capacity to provide sensitive, responsive parenting to their children, despite their best intention to provide the best care for their children. And we also know that less sensitive or negative or unpredictable parenting could put children at risk for immune dysfunction. So mothers' ACEs could potentially impose immunological risk on their children through less sensitive, responsive parenting. So another good news is that parenting is highly modifiable through effective intervention. An attached program, or attachment and child health parenting program, was developed to improve sensitive and responsive parenting for families affected by adversity, such as maternal depression, domestic violence, and low income. And for attached program, they focus on improving parents' reflective functioning, which means parents' ability to think about and identify mental states, thoughts, and feelings and intention in themselves and in their children. So briefly, attached program is comprised of 10-week psychoeducational parenting intervention with dyadic and triadic components to foster parental reflective functioning through practice. So the weekly sessions would include a focus on learning and practicing reflective functioning skills. And the session would include leading by example, asking questions, and providing opportunities to reflect on actual mother-child interactions during the session via videotaped caregiver and child-free play session, followed by a hypothetical and a real-life situation. So there are published papers that have demonstrated positive impacts of attach on parental outcomes, parent-child relationships, and children's health and development. And of interest to today's talk, the group of people have piloted attach have found that mothers and children who completed attach intervention had lower CTRA scores, which indicating a healthier immune cell gene expression profile compared to mother and children in the wheatless control group. So in this pilot analysis, the impact of mother's ACEs on the diet's CTRA profiles in a context of attach has not been conducted. So for our study, our current study, the aim is to determine whether mother's ACEs is associated with increased inflammatory gene expression in mother-child diets. And we hypothesized that higher maternal ACEs could be associated with higher CTRA scores in mother and children. And we also hypothesized that attach could attenuate the association between maternal ACEs and mother-child CTRA scores. So for methods, the study has been approved by University of Calgary IRB, and we recruited 20 mother-child diets from a domestic shelter in Canada, Calgary. And they were randomized to either intervention or a waitlist control group. And mother and children provided one-time dry blood spots after intervention group completed the attached intervention or before the waitlist control families start the intervention. And ACEs were measured by the 10-item original CDC ACE measure. And we also included these covariates in the analysis. So for inflammatory gene expression, the non-fasting blood collected through the DBS card was assayed for genome-wide transcriptional profiles by RNA sequencing. And gene expression data were analyzed using a standard pre-specified set of gene transcription transcripts used in previous research to quantify CTRA RNA profile. And these include, in total, 47 CTRA indicator genes available for analysis. So more methods. For the first aim, we used the marginal effects analysis model 1, which quantified the association between mother's ACEs total score and CTRA scores in mother and children respectively, while controlling for intervention and control condition. And then for the second aim, we used model 2, which added the ACE by intervention group interaction term with a follow-up simple slope analysis quantifying the association of CTRA scores with maternal ACE separately for participants in attached intervention versus control group. So for results, firstly, we didn't find any differences in terms of age between intervention and waitlist control group. And for AIM-1, we found that in mothers, the association between maternal ACE and CTRA scores were marginally significant, such that there was a trend for higher maternal ACEs to be associated with higher maternal CTRA scores. In children, higher maternal ACEs was significantly associated with higher child CTRA scores. And also as a sensitivity test, when we control for covariates, the result remained the same. For second AIM, so in mothers, the interaction term was not significant with or without covariates in the model. So the results here only shows the children's interaction term. So in children, the interaction term was significant. So the blue line indicate control group, while the orange line indicate intervention group. So if you look at the before ACE reached 7, at the same level of ACEs, the ones in the intervention group have lower CTRA score. So that suggests that the association between maternal ACE and child CTRA scores was attenuated for attached intervention group as compared to the waitlist control group. And then we also found, if you just look at the orange line, there is a significant association between maternal ACE and child CTRA, such that lower maternal ACE score were associated with lower child CTRA scores, which suggests that the intervention was more effective for children of mother less affected by early life adversity. So there are many limitations of this study. First of all, we have very small sample size, so spuriousness still remain. And we used very selective sample of mother and child recruited from domestic violence shelter. So they could represent a group that have higher ACEs compared to general population. And as Dr. Dong mentioned earlier, there are many models conceptualizing ACEs or operationalizing ACEs. And for our case, it will only use a total score, not considering the chronicity and severity and when mother have the exposure to ACEs. So in conclusion, in this highly selective, high-risk sample of mother-child diets affected by domestic violence, maternal ACEs were associated with poor child, but not maternal CTRA scores, and the attached parenting intervention conferred more protection in relation to the children when mother's ACEs were low. So our findings suggest that parental ACEs could be transmitted across generations, potentially through this pro-inflammatory skewed immune activity. And the finding also suggests that parental intervention could moderate this intergenerational impact, and the effectiveness of the parenting intervention could vary by parental exposure to early life adversity. And here's the website of ATTACH program if you want to learn more about the intervention, and acknowledgement of the team that made this study possible. And thank you, that's it, and it's my contact. The kids were before age of 6.
Okay.
Yeah.
Just a quick clarifying question. So on your results slide, is it possible to go back to
Yes.
it actually?
Which one you want?
The one that had the orange, the ones with the lines.
Oh, yes.
That one. Yeah. So I'm just trying to understand. Maybe I don't quite understand the interpretation of this, but it looks like the sign's changing for your orange line. Does that mean that this attached parenting program actually made things worse for something? Or I guess, can you help me understand the interpretation of the points in this figure? I don't think I quite understood.
Yeah. So this is showing the interaction effect. So I guess I can go back to this one. So this is showing the effect of attach on mother and child's CTRA score. This is actually looking at the intervention effect. What I'm showing, so in this slide, what they're showing is that compared to the control group, the ones in intervention have better immune cell gene expression profiles, just looking at the intervention effect. What I'm looking at here, so the orange line, so the x-axis is indicating ACE total score, and the y is the CTRA scores. So what the orange line is showing you is as ACE increase, the CTRA score increase. It's showing that higher ACEs, higher pro-inflammatory profile. So what this graph is showing you is comparing different trend in control versus intervention. And what's interesting, I think it could be because of the small sample size, the control group actually don't have any, the slope is not significant, showing that there's actually, in the control group, ACE doesn't have any significant influence on the CTRA score, but for the intervention group, higher ACE, higher CTRA score. So it's kind of contradicting to what we hypothesized. We're hypothesizing that the intervention would attenuate the association, but what it's showing here, it's kind of a competing effect instead of attenuating. So it could be that the mothers with higher ACE score need more resources compared to the ones with lower, which is what the program is intended to do. They are designed to implement with other programs. But if you're looking at before ACE reach 7, at the same level of ACEs, the one in the intervention still have lower CTRA score. Yeah.
Thank you.
Great, thanks very much.
Yeah, thank you.
Hi, everyone. Thank you for being here. Appreciate that. Hi, everyone. My name is Tamkina Tarauf. I'm in the Department of Sociology at UW-Madison. And it is my great pleasure today to introduce our speaker, keynote speaker for the day, Dr. Anya Prince. So I'm a sociologist by training, and I study human well-being, both factors within ourselves and beyond ourselves that influence well-being, so including genomics. And I also study how scientific knowledge about well-being shapes our opinions, choices, and capabilities. So no wonder I was really looking forward to Anya's talk. As genomic discoveries continue at an exponential pace, and today's panels were very much evidence of that, it seems like there's enormous potential for this knowledge to advance human well-being. Yesterday's panel, for those of you who were here yesterday, alluded to some of the best-case scenarios about how this knowledge might be used. So technological advancements such as maybe precision medicine or precision education that can equalize treatment effects across patients and pupils. As a sociologist, I'm also very intrigued by the psychological and cultural transformations that result when a medicalized gaze is cast upon traits that we used to think were maybe of our own choosing. Perhaps this lens informs us to make better decisions. Perhaps it can make us more tolerant of the follies of others. But as yesterday's panel discussion brought to the fore, utopian ideals can sometimes easily devolve into very dystopian realities, threatening to compromise principles that we hold dear, like liberty, equity, and justice, in ways that we may not easily foresee, demanding us to tread carefully and think critically. So today, it is, again, my great pleasure to introduce Dr. Anya Prince. Over the past decade, Dr. Prince has become a leading expert voice in the study of matters related to the ethics of genomics research. She has published dozens of articles about the implications of genomic research on topics ranging from insurance, information disclosure, patient and consumer rights, and more. In 2022, Dr. Prince was awarded an NIH grant to specifically focus on the implications of polygenic indices for social traits, given that the interpretation of these indices in particular pose some unique ethical challenges. We have the privilege of learning today about some of this work, so please join me in welcoming Dr. Anya Prince.
Thank you very much. Although I have to, you've given me a title I don't have, so I'm a Juris Doctor, but not a doctor. So Esquire, if you really want to be pompous. So I'm really excited to be here. So for those of you who don't know about the ELSI field, or the ethical, legal, and social implications field, when the Human Genome Research Project started, people were really worried about the ethical and legal and social implications. And so what NIH did here in the US was actually say for 5% of funding that we spend on genomics research, or of every dollar we spend on genomics research, 5% needs to be set aside for ELSI research. And so there's actually a really robust community of ELSI research, very interdisciplinary, much like the interdisciplinarity of this room, that looks at issues around medical genomics. And so through those grant mechanisms, we applied for a grant to look at the social traits as well. So just a little background into ELSI if you weren't familiar with that field. There are, as sort of the motivation behind this is thinking about a lot of the ELSI researchers to date who are thinking about polygenic scores, particular, are thinking about them in medical traits. So if you were here yesterday, I talked about a big project that's looking about the translation of PGS for heart disease and cancer into the clinic. Right? And that raises a whole host of ELSI issues. But of course, for us all in the room, we're interested in the social part of this. What are the scores that we can create using social and behavioral traits? And so that's the focus for us. And I'll touch on some of the questions that Matthew got about the blurry line in between these, right? This is definitely a continuum and not two silos. And so there's a lot of really important potential benefits of I promised that if I paced, I would carry the microphone. So there's a lot of really important potential benefits of polygenic scores for social traits. That's why you're all here, because you believe that your research is going to be doing good, I'm assuming. Right? But we can help to control for genetics in social science research. We can help genetics research better understand environmental factors. And for some, we can translate that into policy, right? So people say this could be a really good way of having policy informed by both genetics and social science. And of course, there's limitations to this science, as with everything, right? So we've heard about some of it today in people's limitations talks, but the predictive value of these scores might be low. The samples that go into these might be biased. The measurements that we're using, right, how a trait is actually measured or defined might lead to some limitations. There's all sorts of potential limitations that might come with this research, as with all science itself. And then, of course, there are some potential harms from this research. And most specifically, what I think about a lot as a lawyer and public policy person is the harms that could potentially fall when findings from this research move outside of the research setting into society. So we have concerns of genetic determinism. People who say, oh, if I have a polygenic score for high educational attainment, that means that I'm destined for high educational attainment. And what happens if they don't meet that, right? Now, I should note, one of the limitations is that these scores shouldn't be used for individualized assessment, but that doesn't stop that individualized assessment and the harms that might fall from that out in society. We might worry about social actors like insurance companies using this data to say, we have a score for risk tolerance. I'm an auto insurer. One of the measures of risk tolerance is speeding, so why not add risk tolerance into whether or not I'm going to charge you more for my auto insurance? And some might say, sure, why not? That's if it's measuring some sort of association with behavior, that's fine. Others might say, ooh, that starts to feel real icky, right? And so we think about some of these potential harms. So as was mentioned, I'm one of the co-PIs along with Jean Cadigan at UNC in their Department of Social Medicine on a grant, and Matthew, who presented earlier, is on this grant as well. And we are looking at the ELSI issues of this field. And so I'm going to talk about
data from 2 of our aims. We're in year 3 of this grant. And so I'll talk about our horizon scanning, which Matthew talked about a bit. And then we've been doing a lot of data collection around biobank participants and their views, and with both focus groups and surveys. And so I'll give that data, and data that collection that's in process is that we're doing interviews with social scientists whose fields might be impacted but who are not yet using any genetics in their research. And then we're also going to be interviewing polygenic PGS researchers, which is what we're naming the social side, and PRS researchers, which is what we're naming the medical side. And then aim 3 will be about some of the legal and policy implications. So I'll briefly touch on some of that, and happy to go into more hypotheses about that in the Q&A. Okay, so horizon scanning. So this is a methodology that was recommended by the Next Track group, which I'm forgetting their acronym right now, but this is a group that advises NIH on what they should look for into the future and what they should fund. So we pulled this methodology and used it for our project. And so it's Matthew showed some of this, but there's multiple steps to it that includes identifying potential trends and then assessing them. And so this identification is a really robust literature review, as Matthew said. And so we used our search terms that had to do with social, behavioral, genomics, sociogenomics, lots of different combinations of those across lots of different databases, and we identified 983 articles. And then we had 2 people review every single one of those articles and code if it was relevant. And so I'll spend a little bit of time talking about our exclusion, because this came up in questions in the last panel. And so we excluded articles where the main trait of interest was medical. So if they were looking at cancer, we would exclude it, even if one of the things that was helping to predict cancer was a social trait like smoking, right? Or social behavior like smoking. We would still exclude that. And what we did is we had an exclude category, an include category. So there were some that we felt were easy, like income or educational attainment, pretty clear. And then for our team that was coding these, we had an unsure category. And we put those in unsure, and then we discussed those at a team about where we wanted to fall. And so some of the decisions we made there were things that had a DSM code, a diagnostic code, we excluded. So even though there might be some social and behavioral aspects to something like autism or ADHD, we ended up excluding them because it was a diagnosis there. There were some things that fell on the line that we included. So obesity was mentioned, and we actually coded that as a social trait. And that's probably the one that we're most pushing the bounds. But we wanted to look at it because it was just so fascinating in terms of the balance between the medicalization of it and the socialization of it. And so we could definitely critique where we draw the line, because it is a continuum, and I'm happy to have more discussions about that, but that just gives a little bit of sense of what we did. And so that's why there's 560 excluded. Those are the medical, also some animal research that got in there, like the socialization of bees. We're like, no, that's definitely an exclusion. And we ended up with this. And then we coded for many things. I'll start to talk about this. But we looked at the methodology, and we had 2 codes for development and utilization. So development were papers where you all were actually making a polygenic score. So some of the original papers. So for example, the risk tolerance paper that I mentioned, so this would be coded as a development paper because they're actually creating, using GWAS summary statistics, creating the polygenic score. And then for utilization scores, we coded those where they took an already created polygenic score and used it to predict one. So here is a study that looked at things like skydiving and said, does the polygenic score for risk tolerance predict whether or not you are going to skydive? So that's our utilization category. So the data that I'm going to show right now is focusing just on score development and utilization studies, although we have a whole bunch of studies that are commentaries, LC articles, empirical LC articles. So there's other categories that we have in our data set, and I'm just not presenting on that today. So we identified, sort of broadly speaking, 59 sociogenomics traits. So this is obviously not 59. I'm just giving a sampling. And some are forward-looking. So this is the whole trends thing. So something like voting behaviors, I think didn't have a polygenic score at the time we were looking. I should say the literature review was from 2017 to 2022. So we pulled in that 5-year period. So something like voting behaviors at that time didn't have a polygenic score, but was talked about or had a GWAS, so you could potentially develop a score there, and then many that we've sort of talked about today in various ways. Okay, so now we can then start to map trends. And so perhaps unsurprisingly, so this is out of our 275 score development or utilization studies, educational attainment was by far the trait that came up the most. And I should say it's not a one-to-one. Studies could have 20 traits of interest or that are being used to predict. And so you can start to see some of the trends here and some of the ways that we collapsed things. And so this is the first 10, except there's a tie for 10th place with the last, or there's a tie for something of the last 3. I'll have 14, so that's why there's actually more than 10 on the side. Okay, and so then we coded, traits. The other thing that we coded is whether or not the article talked about limitations, harms, and benefits of the research. Right? And so when we look at limitations, these are the most common limitations that were discussed. Now, I should say, just as a limitation of our own study, we had 2 coders for each one, but this is our interpretation of the text that we're reading. And so there is some subjectivity to this because sometimes it's hard to figure out, well, is this really like a small effect size, or is this a predictive power limitation, or is this kind of both, or like, are those all the same thing in this, so some caveats there. But you can see that a really common one that's mentioned is portability across ancestry. Most often that was, this study only has people of European ancestry, and so our findings are limited for that reason. We also have portability across other factors that was brought up less often, but still a very common one, as well as some data limitations and how the traits were measured and the like. And then we looked at how benefits were discussed. And now let me just note here that we're talking out of 275 articles, and it's really important to look at the numbers. So here we have the benefits were at 90 or 80 something was the 80 articles or so talked about how this is beneficial because we can better understand the causes of this trait. Pretty good benefit to put in your article when you're reading that. And then we have, sort of the things. But you'll notice that a lot of the benefits are really research focused, right? The benefit of this is that we are going to have better research. We're going to better understand environmental effects. We're going to better understand gene by environmental effects. But you start to then see some articles that start to push into outside of the research realm and into society. So that's where we start to get things like we can maybe use this to decrease social injustices, right? Have fewer health inequalities, or to better understand health disparities if we can understand differences in gene by environment. And I think we could think about some of the presentations we heard today that might be talking about some of those types of benefits. And now I want you to look at the N, and these are the harms that are discussed. Right? So where we were at 70 or 80 in terms of benefits, very, very, very few of the articles engaged with potential harms of the research. Right? When they did, genetic determinism was the most common thing that was mentioned, as well as possibilities of social injustices that might come, and eugenics or scientific racism, and some other things. Another caveat I should give with this is that we had to make a decision over whether we were going to code something as a limitation or a benefit or a harm, right? And there could be some aligning between a limitation and a harm, and we just made a decision, right? We also coded uses, and so there's a use for like use in a clinic. So some people might say that's beneficial, but if it was just use in a clinic without kind of having a valence, we just coded it as use. And then anyone that talked about use, we coded as that. So there can be some squishiness with this, but I think the trends are very clear. So when we put this together, if you think about the percentage of articles, 80 plus, nearing 90 talked about the limitations. This is unsurprising because there's often a limitation section in the journal, and if you don't have that, the editors are going to say, "Where's your?" Or your reviewer 2 for sure is going to say, "Where's your limitation section?" And then we have benefits, but way less harm. Okay, so as if reading 983 articles on sociogenomic was not enough twice over, we also did the same thing looking at lay media. So we looked at lay search engines like Google or Bloomberg News, and we identified articles and coded them in very similar ways to the academic articles. There were much fewer here. I think that's probably because of the search terms. We use similar search terms, and probably those lay articles aren't always using genetics or genomics, so we might be missing some of the discussion here. But we identified 246, and we included 123. Okay, so I won't talk too much about what benefits, but I want to compare the percentages. So whereas academic, we call it the journal articles academic literature, and the lay, the media, as lay articles. And so you can see, I think the most interesting thing are the harms, right? And so I think one of the takeaways for you all doing this research is, and I'll talk about this in a little bit, you might not feel that it's your place, or sociogenomic researchers in general might not feel like it's appropriate to talk about the harms in the scientific literature. But that is more percentage-wise what the public is thinking about when talking about these results. And so there are implications of that when it's not discussed in the science itself. And that's not to say that sociogenomic researchers aren't talking about harms. That could be in a commentary, right, which I'm not presenting here, but it's not in where the science is developed. And so why some of the difference? I think some of it is comfort level, place for it, right? Like if you start talking about harms in a journal article, the reviewer is going to say, we're not going to publish this at all. I think there's a lot of pressures, and I'll talk about some of that. And just to talk a little bit about the different focus of harms, so I had shown the types of harms that were talked about in the academic literature, and these are the most popular harms that were talked about in the lay literature. And this is N's, right? So keep in mind that there are way more academic articles and way fewer discussion, right? So this isn't percentages, these are just N's itself. But eugenics and scientific racism, social injustices, and genetic determinism were the main concerns in the public dialogue of this research. So how do we think about this? One thing to think about is, are there governance structures that impact how researchers present their work? So I talked a little bit about this, but if you have an application for a grant, there's a section that says, like, what are the impacts of this grant? And you're not going to write, it's going to cause catastrophic social injustices in that, right? IRBs actually don't look at these types of social harms. They look to whether or not there's consent and potential harms to research subjects, but they don't look at results and things that could happen outside of the research realm. Stanford has a really interesting new model where they actually have an IRB and then a second social impact group. I'm forgetting their name. exactly, but you essentially have a review board that thinks about the potential social impacts of research. And so you both go through an IRB review and this like social this, and they're focusing really on AI research there. But I think there's questions of whether that type of governance structure should be available for other types of research as well. And can journals do anything to encourage this discussion? And then I think many of you might be sitting here thinking, should there be, right? Like, no, we don't want to talk about these harms. It's not our responsibility to talk about these harms. And so I think there's a difference of opinions over who should be thinking about these harms and whose responsibility it is to discuss the harms. And so many researchers might think, well, it's not my job to think about how it's going to be used in society, because as long as I do good research, talk about the limitations, then that's all I need to do. But then the companies who are monetizing this data aren't necessarily the ones who are going to think, is this the ethical way of having this go forward? And policymakers might not know that they should be thinking about this. And so I think what happens is that nobody is taking ownership over thinking about how to prevent these harms. But there are many people who, if you ask them, are you worried about this? They might say, yeah, I'm worried that some of this research could lead to social injustices. So how do we think about governance structures or systems that can help to have beneficial research, but minimizing the potential societal harms if these scores are used out in society? Okay, so that's our horizon scanning data. Happy to talk more and questions about that. But I also want to give a little bit of information about some of our empirical data on this. I mean, I guess that was empirical too, but our quantitative and qualitative methods data. So we were really interested in looking at biobank or biobank participant views on this. And so one thing we coded for in our horizon scanning was biobanks. We also coded for funders, so we have a whole thing of who's funding this research. But unsurprisingly, the studies that we've heard about today, Health and Retirement Study, AdHealth, UK Biobank, Wisconsin Longitudinal Study, are the ones that are most commonly cited as data sources that are being used for this research. And so how do we think about this? If anybody is from Los Angeles or has been to Los Angeles, this is an awesome joke. I'm from LA, so anyway, if you don't get it, it's fine. Okay, so broad consent. Why am I talking about broad consent? So when participants join biobanks, they consent to having their data used. And what they do is they give now, under newer regulation, they give what's called broad consent, which means I'm not giving consent. So we used to have a model where you gave consent for every single research study you were part of. And the people had to understand what was being studied, what the potential risks to themselves were, what the potential benefits to themselves were, and all of that. And then we realized that's really putting a hamper on science, because we have all of this data and we have to keep on going back and back and back and back. So we came up with broad consent, which is saying, can we keep your samples and then use it for future research with it de-identified or identified depending on the consent, and use your data over and over, and you might not know the specifics of what's studied. And so all of these biobanks, the reason you have hundreds of thousands of millions of data points that you all can use for your research is because at some point, those people gave broad consent, presumably, for a lot of these biobanks. Right? And so just an example, this is the UK Biobank broad consent. And it says, I give permission for long-term storage and use of my blood and urine samples for health-related research purposes, even after my death, and relinquish all my rights. So what do you think I focus on here? Right? They're giving broad consent to health-related research. And there is a lot of data and research studies that are going on social traits. And now that might have really important impacts on health, but is that what the participants were thinking of when they consented to this? And the most provocative example that I always use is that the research study creating the polygenic score for same-sex sexual behavior used UK Biobank data. So if you went back and interviewed UK Biobank participants and said, here's the broad consent you signed, Here's a study on same-sex sexual behavior. Did you think that that is what you gave permission for your data to be used? I'm going to guess that a lot of people would say no. And if you asked a further question and said, would you have given consent for that study? There's probably not an insignificant portion of people who would say, no, I would not have consented to that study. And so I think that's a really important thing to think about when using this, even if it's de-identified, right? So some people might say, so what? It's de-identified. There's no harm to this particular person. But if we think really, if we care about people's participation in research that's voluntary, that is ethical, right? I think it matters, even if it's de-identified, what they think they're giving their samples for. And so we then wanted to know, right? I just gave you a hypothetical of what I think might happen if we asked UK Biobank participants. We didn't have access to UK Biobank participants, but we did have access to Mayo Clinic Biobank participants. And so we both surveyed Mayo Clinic Biobank participants, 467 of them, and then we had focus groups with 66 participants across 11 different focus groups, 5 virtual and 6 in person in 2 different locations, 1 in Florida, 1 in Minnesota. And so I'll present some of this data. So these are the Mayo Clinic, this is our study, the demographics. Unsurprisingly, it skews white because this is the predominantly Mayo Clinic Minnesotan data. Okay, and it also skews higher education and older. Okay. We also did a Qualtrics survey because we wanted a more diverse group. So this is just the general public. We don't know whether or not they are biobank participants. Some probably are. Some who are probably don't realize they are. And so here we have a much more diverse population because we controlled for that. We specifically sampled for that. Okay. So here are some questions that we asked them. So we asked, I trust scientists to think about social implications of their research. Good news. Most people say, yeah, I trust people to think about that. And I trust scientists to think about risks or harms of their research. And many said yes. Now we didn't ask, I think that these questions are interesting when thinking about the horizon scanning data about actually then, scientists might be thinking about the risks and harms, but then how is that communicated and what does that mean for trust of the scientific endeavors? We also asked about whether or not people supported research on polygenic scores for medical traits versus social traits. And so here you will see that both in the Mayo Clinic biobank participants and the general public, there was much more endorsement of feeling comfortable about research on polygenic scores for medical traits than for social traits. And when talked about whether or not they felt comfortable with their information being used to develop these scores, there was much higher endorsement about being comfortable with their data being used for medical traits versus social traits. And so going back to what might they have been giving broad consent for, this helps to back up the hypothesis that maybe they wouldn't be as comfortable with some of this. But we can, through our focus groups, we can add some more gloss to this. And so generally focus groups participants, and we pulled the focus group people from the survey. So all of these focus group participants had taken our survey. So they were generally more supportive of the use of their data to develop scores for medical traits than social traits, but they were really pretty supportive of biobanks in general. And I'll show you some quotes for that. But they seemed mostly worried about what happens when the research goes outside of research. So here's a quote for medical versus social. For me, I would have no problem whatsoever if the genetic information from what I donated was used in medical research if I could help someone. As far as the social traits, I'm on the fence. I can see tremendous benefit and I can see tremendous negatives of that. Here's a quote for trust in biobanks. This is positive research. I trust the clinic. I trust what the data is going to be used for. Scientific research here, it isn't something, I don't believe you guys have a negative intent here. You're not going to go and weaponize it. This will be used, I think, to expand our knowledge in the future of our children and our society and our culture. So really trusting who is doing this research. In terms of use of data, we have people really acknowledging they signed up for this. So for research purposes, I guess I still hold true to when I first initially consented that this is what I signed up for. So I'm okay with it because of that. The biobank that I volunteered for is in providing my genetic material data for future research. I trusted them as good stewards. Similarly, I'm a pretty open book. I would have no issue with it. If the scientific community would find value in it, I've got no problem with it. But then we see on the other side some concerns about what happens outside. So we obviously all care enough about research that we participated in the biobank to begin with. And we followed up with and answered the survey. And then we said, yeah, we would do this. Even science has a way of sometimes going off the rails. Oppenheimer was a big movie over the summer. We all, everybody heard about it. Everybody knew he did the thing back during World War II. So if it's just for the sake of research, gaining knowledge and gaining insight into the human condition, but when you let sciences use some stuff. Okay, I'll skip that for time. Okay, this is where then I come back to the biobank participants generally were really supportive of this for research, but nervous about what happened outside, and we have more quotes about that type of thing. So how do we think about that? And so we have all sorts of legal structures out here that constrain or don't constrain use of genetic information. And we have laws in place. These are sort of the common areas where maybe social genetics might be used. And we have laws in these spaces, and I'm happy to talk about any of these in Q&A, that could theoretically prevent some uses, but there's lots of gaps in them, right? So there's a genetic discrimination law that prevents use by employers and insurers, but it's only health insurers. It's not life insurers. The commercial space is sort of a wild, wild west, right? The FDA could theoretically regulate, but then again, we come back to the medical social line. The FDA has purview to regulate medical devices that deal with conditions and diseases. I think it's an open legal question whether the FDA would have actual purview to look at some sort of a social trait score that is somehow commercialized because if it's not medical, right? So I think this challenge some of our legal structures in place so we can talk about how we really need to have a law and a legal system that prevents some of the bad uses, but our legal structure is definitely not there yet. And so I think a lot of, going back to the discussion of harms, I think there needs to be a greater discussion over what the potential harms could be in order to have robust policy that fills in some of the gaps of these legal frameworks. So I'm happy to take questions. Thank you to everybody who, especially all of the Horizon Scanning Data team who read lots and lots of your all's articles. So, thank you.
Thank you. I loved your talk. I have about 50 questions, but I'll narrow just to one, which is building from the Horizons work, which thank you for introducing us to I hadn't heard about that before, so it's incredibly cool. Makes me think that so many of our research articles are written in a way that's so formulaic, particularly introduction sections. And so I'm sort of wondering about within the constraints of how scientists are often trained and the ways that our articles are organized, what would it look like if we were to start to think about adding in more details with respect to the harms? Because it's almost like in the introduction you would say, so the formula is we have this problem, we know a little bit about it, prior studies are limited, my study is going to solve this by doing this. But caveat, we probably shouldn't do this study, or this is controversial to do that study, or these are the harms of doing this study. But we did it anyways, and so here are the methods for how so I'm just kind of thinking like very practically, right? Because to some extent, we can add it to the discussion, and that's an easy place for us to do it, and I think we should. But I'm sort of just wondering, like, should the formula change for how we publish work in sociogenomics and have different standards for how our papers are organized and structured? So I'd love your thoughts on that.
Yeah, great. So a couple of things. So one, in terms of just should this research be done at all, right? I think there's probably some research that I might say, oh, no. But there's a lot of research where the harm comes from misinterpretation or overinterpretation. And so some of it is just calling that out, right? A reminder, these are for the general population. These are not individually specific scores. So one effort that has been put out by some of the groups is to do FAQ documents. And so these are documents that kind of go through what is a potential harm of this? What are some potential implications? How might this data be misused? So it's a really positive way of doing that. One critique I have is that sometimes those are in the supplement and they are not mentioned in the article themselves. And so if I'm a naive person coming to scientific research, I'm not going to read your supplemental figures and data and try to find it. So just put a note in the discussion of this has implications, see supplement, here's this FAQ. And there's a whole standard format to talk about some of those things. If in even more engaged approach that some researchers do, and if the journal allows it, and this is where I think the journals have some onus here, is to allow like a box, right, a figure that says, here are some of the limitations or caveats that we should think about if this were to be used in society. So this is really beneficial in the research context for these reasons, and this would be if used in this way or that way, right, or this isn't how it should be used. And so like the same-sex sexual behavior paper did things like that, where they really talked about this shouldn't be used for discrimination. This isn't actually measuring whether somebody is gay or not. It's measuring, right? And so they just did some caveats on what actually is being measured, what's the predictive value, how it can be used. But I think I would just say I think it's really important for that to be in the scientific literature itself, right? Or at least flagged there so that you can go find it easily and not just pigeoned off somewhere as like, oh yeah, we filled out that FAQ, but we're not going to talk about it in our study. Patrick's like, the face makes me a little.
I was just thinking about kind of what you're saying here, and I'm thinking about who the audience is of these different things. So I don't expect the average person to look at the academic paper. I presume they're going to consume it through popular press lens or things like that. And so given that, it seems like the solutions of how you mitigate harms or communicate potential harms would be different than sticking, like, it's you have limited words in the paper and putting stuff in the paper that's not going to be read by, I do wonder what like the benefit is going to be of putting it someplace like that.
Yeah, I mean, I think that's definitely fair. I would say I don't think this is the only fix, right? I don't think that if we talk about potential societal harms or potential misunderstandings that could come from this data, that all of a sudden there won't be bad uses out there in society. But I think a first step is tying it to the research itself. If only just so that the next researcher who looks at it thinks about it. Like one of my critiques about just focusing on the limitations is it kicks the can down the road, right? The GWAS study says, we didn't use European ancestry. That's our limitation. Kick. And then the utilization study says, our summary statistics didn't use European ancestry. Kick, right? And so by having just limitations, it just kind of allows that to always be like, well, it's not my problem. That's the data, right? So some of it, I think, goes back to the responsibility part. But I would also say, where are the journalists getting the information? Like, at some point, there might be somebody reading that article, and I don't think this has to be 800 words, right? There could be a box that's 200 words that talks about, or an FAQ document that then you could also hand to journalists, right? That could have that dual use. And then in a most extreme example, I think some of the scientific literature is being wildly misused, but in the tragic Buffalo shooter case, there was the manifesto of the shooter. Now, this is, of course, not like how many people consume science, but some sociogenomic science was cited in that to justify incorrectly, right, the racist ideologies that led to that shooting. And so would a box have fixed that? No, probably not. But like, at least maybe then I think it's still important to connect those two if it's cited, that it just would put a grain in somebody's mind to say, oh, maybe I should think about this.
Hi, yeah, thanks for your talk highlighting what the public thinks is some of the ethical problems. With the UK Biobank, you're discussing the ethical and legal problems of using their data for non-health related measures. Setting perhaps the ethical part aside, do you think it's not legal to use their data? I mean, there's a second clause in there that said they waived all their rights. So do you think that like gets you off the hook legally? Or if you're American, are you off the hook anyway, not British? Or how does that play into it, do you think?
Yeah, so I don't know about the like UK legal context. I mean, I think one thing, I think less about the researchers using that data and more of the biobank who is approving that data, right? In order to use that data, you're putting in an application, is my understanding of it. Although some of the summary statistics, maybe not, so you can all correct me if you're, but I think often you're like putting in an application to say, can I get this data? And so I think that's on the biobank governance to say, is this what our participants understood? Because I just pulled out that quote. Who knows what the dialogue was like, or how the studies are being pitched? But I think it's I think that's more of a biobank governance side than an individual research side. But I do think it's important to think about, even if it's an anonymous data, what did the participants want? So I was having a conversation with Q earlier about imputing data, right? So if you're imputing data that somebody didn't fill out a survey question on, did they not put the survey question, fill it out because they were worried about stigma, because they were told you can skip any question you want on this social science research because you are voluntarily participating in this research, and then on the back end we're filling in the data. I mean, I think these are questions that are fair to ask the field so that we're respecting people's rights. But is it like illegal to do this? No, I don't think so. I think it's just questions of respect. Or let's change the broad consent going forward so that people understand really how their data might be used.
Thank you.
Thanks very much for the talk, and I completely agree with the points made about consent and whether researchers should think more about respecting the initial intention of these participants. But I do wonder, in recent history, has there ever been a case where the scientific community thought, well, we have to come together and decide if this type of research should or should not be allowed, and a decision was made, and who made that decision? I wonder if there has ever been a precedent to this. And I guess the background thought that led me to ask this question was, we have a study going on which is not supposed to be controversial. It's just the shared genetics between autism and ADHD, right? And I was surprised by, I was educated on issues related to this topic. Apparently, some of the studies people did led to a news article that basically said something that this can potentially lead to a cure for autism in the long term, right? But then that really caused a lot of trouble, because apparently there's a community for parents who have autistic kids. They strongly oppose this type of view because they think there's nothing to cure. This is neurodiversity. But when there's this divergence between the scientific effort and possible stakeholders, in this case could be parents or families with autistic kids, who should be the people to make the decision? I mean, I respect the thoughts that, well, if you're using your study to research something I didn't give consent to, they can drop out the study. I think there's a right for people to do that. But then if they did give consent, should the purpose of the study, or should the direction of the study be determined by study participants? That becomes a gray area that I don't have an answer for. I wonder if you can comment on this.
Yeah, great. So in terms of have we ever stopped research before, I mean, two examples that I can think of would be, speaking of Oppenheimer, right? nuclear armament and things. Like there was some discussion, obviously we created the bomb, but there is some discussion there about like, should we be doing this, right? So that's outside of the genetics context. But in the genetics context, there was also the similar conference that was the gene editing, right? And so they actually came together early on and said, should we be doing gene editing, and is there a line for that? And they came up with principles there that we shouldn't be doing gene editing in embryonic tissue. And so now that line nowadays is being pushed with CRISPR and things. And so, but that was an example of early researchers in the field coming together, discussing the pros and cons, and setting some bounds on the research.
Just very quickly follow up. Who made the decision? Was it researchers or was it someone else?
That was researchers that came together. But in terms of your question, I don't want to come across as saying that, you know, if I had my druthers, I'd stop all of your research tomorrow, right? Like, I think that it's less about should we do this research or not, and more about how do we communicate it. And the example of the ADHD autism study is a really interesting one, right? Because the problem there might not have been that we're studying the genetics of autism, not your study, but the previous study, right? But that it was communicated in a way that medicalized people's identity in a way that they didn't like. And so that's where community-engaged research can be really beneficial. If you're doing research on some identity-based trait, speak to people with those traits, right? And see, how do you feel about this research? How would you want it to be communicated? What do you see as the benefits? What do you see as the harms? And that then can go into the documents and into the discussion, and the language that's used can be more respectful. Now, part of the governance structures part is we need grants to also fund that, right? Community-engaged research is very expensive to do that, but not impossible. But like we used some community-engaged research in our project that was extremely helpful. So one part of this is that we took our 59 traits, and we wanted to specifically study 5 traits in our surveys. And so we went to community advisory boards, and we said, of these, which ones do you think we should study? And we came up with a methodology to help get that values elicitation. And so that was extremely helpful, because now we have decided on these 5 to study, and it comes in part from the community of biobank participants who say this is what we want. Now, there's limitations to community-engaged research, but I think, you know, that's the methodologies. But I think that's a way of saying, not should we be doing this research or not, but how do we, I mean, you can do community engagement with that question, but even absent that question, just how should we communicate this, and what are some of the risks and benefits of communicating this way?
Thank you. Yeah, this was a really fascinating talk, and it's really interesting to hear from the focus groups. I don't know if I missed like how you were asking these questions to the focus groups, but I think the question that I'm still left with from there is how do people who took the survey or people who participated in the focus groups understand the definition of health-related research? Do they see something that speaks to social causes of obesity as something that's health-related, that sort of thing?
Yeah, so in the survey, we asked, we had a series of like 10 or 13 traits that range from medical to social, so height and cancer and obesity and income, and we asked them to rate how much genes or environment impacted this. I forget the exact question. So we do have a metric where we could categorize, quantify people on sort of I'm more of a nature person or I'm more of a nurture person, based on where they land there, and then we can then assess how they view the medical and social. And then what we did for that with the focus groups is the very first question we asked them in order to kind of warm up the room is, what do you think you inherited, or what do you think that you got from your family? And then people would say, oh, I got my love of love of cats from my family. And then we said, well, do you think that's more genetic or more environment? And then they had that. So we have really robust quotes from the focus groups themselves about, oh, well, that probably is more like social because there were all the cats around, or like, other ones that were more medicalized. I've been ignoring this side of the room for a little while. Hello.
Thank you. That was a fascinating talk. Thank you for that. I'm curious, and maybe I don't know how much you know about or your expertise in GINA, kind of how deep that goes, but really interested in talking more to a lawyer about GINA. And if you know, why did the architects leave out some of these other types of insurance? If you have any insight on that,
yep.
If there's any movement to do so,
yep.
And it is kind of a relatively new law, I understand, and that there haven't been many challenges to it. I don't know if that's true, or if people have tried to challenge GINA, if there have been lawsuits, if there's, so I'd just be really interested to hear that and whether you foresee any changes or any movement there.
Yeah. Great. So, so GINA is the Genetic Information Non-Discrimination Act. It is a federal law that prevents health insurers and employers from using genetic information. Louis Schlatter, who's the representative that first introduced GINA, introduced it 13 years before it passed. And so there were 13 years of iteration through Congress, some of which included life, long-term care, and disability insurers that did not get passed. And then they dropped those and were left with health insurance and employment. So people were thinking about these, and just politically they couldn't get it done.
So it was like a lobbying thing.
It was a little bit of a lobbying thing, yeah. And then in terms of is there movement, so insurance is regulated at the state level. And so there is movement at the state level to regulate this. So if you all are interested, I had an article that came out, a commentary that came out in JAMA where I'm actually tracking these and looking at how many states have introduced bills that are discrimination, like anti-discrimination in the context of life, long-term care, and disabilities. And a remarkable amount of states across political spectrums, across rural, across everything, have introduced these bills, and the insurance lobby is strong, and they don't necessarily pass. Florida, however, in 2019, 2020, became the first state to ban life insurers from using genetic information. So we're going to see what happens to the insurance market in Florida. The one caveat I will say with that is many times the state laws define genetic information in a way that says that it is like DNA that is linked to a condition or a disease. And so you might actually not have some of the social traits count as genetic information under the law if they're not predicting a disease or condition. And then the last question you had was about challenges to GINA. So in the health insurance space, there's actually not a private right of action, and the Affordable Care Act came 2 years after GINA and completely changed how we underwrite health insurance. So GINA, although it made big waves there, it didn't really have an impact in the health insurance field because the Affordable Care Act did the work for it. For employment, there haven't been that many cases. And what's interesting in employment is the cases that have been brought have been predominantly around employer collection of family medical histoy. Because under GINA, genetic information is defined not just as DNA, but also your family medical history. Do I have time for a really brief anecdote?
Yeah, sure.
Okay, this is a fun one. The very first jury trial for GINA is called the devious defecator case. And it is a warehouse where somebody was defecating in the warehouse, and they wanted to figure out who it was. So they did genetic testing on the sample and then genotyped their employees to try to find a match. They did not find a match, but they got a violation of GINA, and the jury awarded them like $2.5 million, which eventually was dropped down. But that's our first GINA case, which is not what the people were thinking about when they passed the law. Does that mean that genetic discrimination isn't happening? I don't think genetic discrimination in employment is widely happening, but could it? And just the fear that people have over it happening, I think, is the bigger impact. Okay, sorry, that's just a fun one to talk about.
We're right at time, but maybe one more question.
I'll be at dinner too. If people want to keep on chatting.
Thank you. That was fascinating. So you mentioned, like, we were talking about the onus on researchers, right? And you mentioned how when it comes to commercialization and business, it's the wild, wild west. So, and I feel like also sometimes researchers may not be able to oversee some of the harms and some of the ways it may be misused. So I wonder why we don't talk more about the lack of a public discourse, a discussion for like society as a whole deciding those are going to be the guardrails instead of like putting all the onus on the researcher as well. Why don't we get there? Why that never happens? And we would have guardrails as well for business and so on and so forth.
Absolutely. A really great point. So I think part of the reason I'm focusing on researchers is because I'm talking to a bunch of researchers. If I was talking to a bunch of policymakers, I would say, get on this. Look how you're not doing your job and passing laws. If I was talking to a bunch of owners of a commercial thing, I would say, just because you can sell it doesn't mean you should, right? And so I don't think it is only researchers' responsibility because there's a lot to fix. But I do think that if everybody says, not it, it's not my job, then nobody's left actually taking on. So I think it's important for everybody to play a role, and one of the ways that researchers can play a role is engaging in this dialogue so that the public knows and has the ability to have their voices heard over how they want this to happen, and that we have policymakers passing laws and the like. So I think it's one piece of the puzzle, but not the sole piece. Okay. Thank you for having me.